The human nervous system is perhaps the most remarkable communication network ever conceived by nature. Every thought, sensation, movement, and memory relies on signals racing through a vast web of specialized cells at speeds that would make any telecommunications engineer envious. These signals, carried by neurons through electrical and chemical means, coordinate everything from your heartbeat to your ability to read these words. Understanding how these signals work, how they're generated, transmitted, modified, and interpreted offers insight into what makes us conscious, mobile, and alive. What follows is a detailed exploration of neural signaling, from the molecular machinery inside a single neuron to the complex circuits that govern behavior, presented at a pace designed for contemplation or rest. To understand how the nervous system sends signals, we must begin with the neuron itself, the fundamental unit of neural communication. These cells, specialized beyond almost any other in the body, have evolved a distinctive architecture that reflects their primary function: receiving, processing, and transmitting information across distances that can span from fractions of a millimeter to well over a meter in length. A neuron is not simply a blob of cytoplasm with a nucleus, though it certainly contains these features. Rather, it is a highly polarized cell with distinct regions, each contributing to the overall process of signaling. At the heart of the neuron lies the soma, or cell body, a roughly spherical structure that houses the nucleus and the majority of the cell's biosynthetic machinery. The soma typically measures between 10 and 25 micrometers in diameter, though this can vary considerably depending on the type of neuron. Within this compact space resides the nucleus, containing the cell's genetic material, surrounded by rough endoplasmic reticulum studded with ribosomes, engaged in the continuous production of proteins. The Golgi apparatus sorts and packages these proteins, many of which will be transported to distant parts of the cell. Mitochondria, scattered throughout the soma, provide the substantial energy requirements of these metabolically active cells. Extending from the soma are two types of processes that give neurons their characteristic appearance. Dendrites, typically multiple in number, branch extensively from the cell body, creating what is aptly termed a dendritic tree. These structures serve as the primary receiving stations for incoming signals. The branching pattern of dendrites can be extraordinarily complex, with some neurons possessing dendritic arbors that span hundreds of micrometers and contain thousands of branch points. The surface of dendrites is not smooth but rather studded with small protrusions called dendritic spines, specialized structures that serve as the postsynaptic sites for most excitatory synapses in the brain. These spines can number in the tens of thousands on a single neuron, each representing a potential point of contact with another cell. The geometry of the dendritic tree is far from arbitrary. The branching pattern, the density of spines, and even the electrical properties of the dendritic membrane all contribute to how the neuron integrates incoming signals. Some dendrites are relatively straight and unbranched, while others create elaborate candelabra-like structures. The total surface area provided by the dendritic tree can exceed that of the soma by a factor of 10 or more, dramatically increasing the neuron's capacity to receive inputs from other cells. The axon, in contrast to the typically multiple dendrites, is usually singular, extending from a specialized region of the soma called the axon hillock. This is where, as we shall see in later chapters, the critical decision to generate an action potential is made. The axon itself is a thin, cable-like projection that can vary enormously in length. Some axons, such as those connecting adjacent neurons in the cerebral cortex, may be only a few tens of micrometers long. Others, like those running from the spinal cord to the muscles of the foot, can exceed a meter in length in an adult human. To put this in perspective, if we were to scale up a typical spinal motor neuron to have a soma the size of a baseball, its axon would extend more than half a mile. The axon maintains a relatively constant diameter along most of its length, typically ranging from a fraction of a micrometer to over 20 micrometers in the largest axons. This diameter is not inconsequential, as we shall discover when discussing signal propagation. The axon's structure is maintained by an internal cytoskeleton composed primarily of neurofilaments and microtubules, which run parallel to the axon's length. These provide both structural support and serve as tracks along which molecular motors transport cargo between the soma and the axon terminal. At its distal end, the axon typically branches into multiple terminals, also called synaptic boutons or axon terminals. Each terminal represents a specialized structure designed for the release of chemical messengers. A single axon may give rise to hundreds or even thousands of terminals, allowing one neuron to communicate with many others simultaneously. Within each terminal, we find a high concentration of synaptic vesicles, small membrane-bound spheres filled with neurotransmitter molecules. These vesicles cluster near specialized regions of the terminal membrane called active zones, where they will eventually fuse and release their contents. The transition from axon hillock to axon proper represents a significant change in the cell's composition. The axon hillock has an unusually high density of voltage-gated sodium channels, making it the most electrically excitable part of the neuron. This is no accident, as this region serves as the integration point where all the various inputs received by the dendrites and soma are summed to determine whether the neuron will fire an action potential. Supporting the axon along its length, we often find a specialized wrapping of membrane called the myelin sheath, though not all axons are myelinated. This insulation, produced by glial cells, dramatically alters the electrical properties of the axon and increases the speed of signal transmission. We shall explore this in detail in a later chapter, but for now it suffices to note that myelination represents one of the key innovations that allowed nervous systems to evolve rapid, long-distance communication. The membrane that encloses all these neuronal structures deserves particular attention, for it is here that much of the signaling magic occurs. The neuronal membrane is a lipid bilayer approximately 7 to 10 nanometers thick, composed primarily of phospholipids, arranged with their hydrophobic tails facing inward and their hydrophilic heads facing outward toward the aqueous environments inside and outside the cell. Embedded within this bilayer are numerous proteins, ion channels that allow specific ions to cross the membrane, pumps that actively transport ions against their concentration gradients, and receptors that respond to chemical signals from other cells. The composition of this membrane is not uniform across the neuron. Different regions express different combinations of channels and receptors, creating functional specialization. The dendrites are rich in receptors for neurotransmitters and certain types of ion channels that support integration of inputs. The axon hillock concentrates voltage-gated sodium channels. The axon terminal is studded with voltage-gated calcium channels essential for neurotransmitter release. This regional specialization reflects millions of years of evolutionary refinement. Neurons come in a remarkable variety of shapes and sizes, reflecting the diversity of their functions. Unipolar neurons, found primarily in the peripheral sensory system, have a single process that branches into two, one extending to the periphery and one to the central nervous system. Bipolar neurons, common in sensory systems like the retina, have two processes extending from opposite poles of the soma. Multipolar neurons, the most common type in the brain, have multiple dendrites and a single axon, creating a star-like appearance. The Purkinje cells of the cerebellum have some of the most elaborate dendritic trees in the nervous system, with branches so dense they appear almost two-dimensional. Pyramidal neurons of the cerebral cortex feature a characteristic pyramid-shaped soma and an apical dendrite that extends toward the brain's surface. Within the neuron's interior, beyond the major organelles of the soma, exists a complex cytoskeleton that maintains cell shape and enables intracellular transport. Three types of filaments compose this skeleton. Microtubules, approximately 25 nanometers in diameter, serve as highways for molecular motors carrying cargo. Neurofilaments, intermediate filaments specific to neurons, provide structural support, and actin filaments, thinner at about 7 nanometers, are particularly important in dendrites and dendritic spines, where they support structural plasticity. The transport system within neurons represents a considerable logistical challenge. The soma contains the biosynthetic machinery, but proteins and organelles are needed throughout the cell, including at distant axon terminals. Two types of transport solve this problem. Anterograde transport carries materials from soma to terminals, powered by kinesin motor proteins walking along microtubules. This transport moves at speeds of 1 to 400 millimeters per day, depending on the cargo. Retrograde transport carries materials from the periphery back to the soma, powered by dynein motors moving at similar speeds. This bidirectional traffic is essential for maintaining the neuron's far-flung empire of processes. Neurons are also characterized by their tremendous metabolic demands. Though the brain represents only about 2% of body weight in humans, it consumes roughly 20% of the body's oxygen and glucose. Much of this energy expenditure goes toward maintaining the ionic gradients across the neuronal membrane, gradients that are continuously dissipated during signaling and must be constantly restored. The sodium-potassium pump, an enzyme that spans the membrane, works ceaselessly to exchange sodium ions inside the cell for potassium ions outside, consuming ATP in the process. This pump is so important that it accounts for perhaps half of all the energy used by the brain. The neuron's reliance on continuous energy supply makes it particularly vulnerable to disruptions in blood flow. Unlike muscle cells, which can tolerate brief periods of oxygen deprivation by switching to anaerobic metabolism, neurons have very limited capacity for anaerobic ATP production. This is why strokes, which interrupt blood flow to brain regions, cause such rapid and devastating damage. Neurons begin dying within minutes of losing their blood supply, unable to maintain their ionic gradients, and experiencing a cascade of damaging events, including excessive calcium influx and excitotoxicity. The diversity of neuronal form reflects diversity of function. Motor neurons, which command muscles to contract, have long axons extending from spinal cord to periphery and relatively small dendritic trees as they integrate inputs from a limited number of sources. Sensory neurons, which bring information from the periphery to the central nervous system, often have specialized receptor endings rather than typical dendrites. Interneurons, which make up the vast majority of neurons in the brain, show enormous variety in their morphology, reflecting their diverse roles in processing information. Some are designed for precise local inhibition with compact arbors. Others have axons that spread widely, broadcasting signals to many targets simultaneously. As we consider this elaborate architecture, we begin to appreciate that form and function are intimately related. The extensive dendritic tree of a cortical pyramidal neuron reflects its role in integrating thousands of inputs from diverse sources. The long axon of a motor neuron reflects the need to span the distance from spinal cord to muscle. The thousands of terminals arising from a single axon reflect the neuron's role in influencing many downstream partners. Each structural feature has been shaped by selective pressure to support the neuron's role in the larger network. Yet all of this structural complexity would be meaningless without the electrical properties that make neurons excitable. The elaborate morphology of dendrites and axons is not simply scaffolding, it creates specific patterns of electrical signal flow. The particular mix of ion channels expressed in different parts of the neuron determines how signals are shaped and transformed as they travel. The relationship between structure and electrical function is so intimate that changes in one inevitably affect the other. When synapses strengthen during learning, they often do so through structural changes, adding receptors and even growing larger. When neurons are injured, the disruption of electrical signaling leads to structural degeneration. This brings us naturally to the question that will occupy our attention in the next chapter. How do these structural features create the electrical properties that make neurons capable of signaling? What is the nature of the electrical state of a neuron at rest before any signal has been generated? To answer this, we must examine the distribution of ions across the neuronal membrane and the forces that govern their movement, establishing what is known as the resting membrane potential. The neuron at rest is anything but inactive. Though no signals are being sent, no action potentials racing down the axon, the cell exists in a state of carefully maintained electrical polarization. This baseline electrical state, called the resting membrane potential, represents the voltage difference between the inside and outside of the cell. In most neurons, this potential sits at approximately negative 70 millivolts, meaning the inside of the cell is 70 millivolts more negative than the outside. This might seem like a small number, but given that the membrane separating these two compartments is only about 7 nanometers thick, this voltage difference translates to an electrical field of roughly 10 million volts per meter, comparable to the field strength in a lightning bolt. Though operating at a far smaller scale and in a far more controlled manner, this voltage difference does not arise by magic, nor does it simply appear when the neuron forms. Rather, it is the consequence of an unequal distribution of ions across the membrane, combined with the selective permeability of that membrane to different ions. To understand the resting potential, we must first consider the major ions involved and their distribution in the intracellular and extracellular spaces. Sodium ions, carrying a single positive charge, are far more concentrated outside the neuron than inside. The extracellular concentration of sodium typically sits around 140 millimolar, while the intracellular concentration hovers near 12 millimolar, a ratio of more than 10 to 1. Potassium ions, also carrying a single positive charge, show the opposite pattern. They are highly concentrated inside the cell at approximately 140 millimolar, while outside the concentration is only about 5 millimolar, creating a nearly 30-fold concentration gradient in the opposite direction to sodium. Chloride ions, negatively charged, are more concentrated outside the cell than inside, with extracellular concentrations around 120 millimolar and intracellular concentrations varying but typically lower. Finally, there are large negatively charged proteins and organic molecules inside the cell that cannot cross the membrane at all. These contribute significantly to the negative charge inside the cell. The presence of these concentration gradients raises an immediate question. Why don't the ions simply diffuse down their gradients until concentrations equalize? The answer lies in the selective permeability of the membrane. The lipid bilayer itself is largely impermeable to charged particles. Ions can only cross through specialized protein channels that span the membrane. At rest, the membrane is permeable to potassium through leak channels, proteins that allow potassium to pass relatively freely. The membrane is far less permeable to sodium and moderately permeable to chloride. This differential permeability is crucial. Consider what happens when potassium channels open. Potassium ions, far more concentrated inside the cell, begin to flow outward down their concentration gradient. As positively charged potassium leaves, the inside of the cell becomes more negative. However, this movement of charge creates an electrical gradient, with the inside becoming negative relative to the outside. This electrical gradient opposes further outward movement of potassium. Eventually, an equilibrium is reached where the chemical force pushing potassium outward is exactly balanced by the electrical force pulling it inward. The voltage at which this equilibrium occurs is called the equilibrium potential for potassium. We can calculate this equilibrium potential using the Nernst equation, which relates the concentration gradient of an ion to its equilibrium potential. For potassium, given the typical concentration gradient, the equilibrium potential works out to approximately negative 90 millivolts. This means that if the membrane were permeable only to potassium, the resting potential would be negative 90 millivolts, the point at which no net movement of potassium occurs. For sodium, with its concentration gradient in the opposite direction, the equilibrium potential is positive, approximately plus 60 millivolts. At this voltage, the inward driving chemical gradient would be exactly balanced by an outward driving electrical gradient. For chloride, the equilibrium potential is typically near the resting potential itself, around negative 70 millivolts, though this can vary. The actual resting potential, around negative 70 millivolts, is neither the potassium equilibrium potential nor the sodium equilibrium potential, but somewhere in between, weighted toward potassium because the membrane is much more permeable to potassium than to sodium at rest. We can think of the membrane potential as a weighted average of the equilibrium potentials of the ions to which it is permeable, with the weighting determined by the relative permeabilities. This relationship is captured more precisely by the Goldman-Hodgkin-Katz equation, which takes into account the permeabilities and concentrations of multiple ion species simultaneously. The question arises, if sodium is constantly leaking in down its electrochemical gradient and potassium constantly leaking out, won't the concentration gradients eventually dissipate? Indeed, they would, were it not for an active mechanism that continuously works to restore them. This is the sodium-potassium pump, more formally known as sodium-potassium ATPase. This remarkable molecular machine spans the membrane and uses energy from ATP hydrolysis to transport ions against their concentration gradients. For each ATP molecule consumed, the pump moves 3 sodium ions out of the cell and 2 potassium ions into the cell. The sodium-potassium pump is electrogenic, meaning it contributes directly to the membrane potential. By moving 3 positive charges out for every 2 moved in, it makes the inside of the cell slightly more negative than it would otherwise be. However, the pump's primary contribution to the resting potential is indirect. By maintaining the concentration gradients, it enables the differential permeability mechanism described above to generate and maintain the resting potential. The importance of this pump cannot be overstated. It is estimated that in neurons, maintaining ionic gradients accounts for a substantial fraction of the cell's energy budget. The pump molecules are densely packed in the neuronal membrane, with millions per cell continuously working to counteract the leak of ions down their gradients. If the pump is inhibited, as occurs when cells are poisoned with ouabain, which binds to and blocks the sodium potassium ATPase, the concentration gradients begin to collapse, and with them, the resting potential. The resting potential, once established, is remarkably stable under normal conditions. The membrane acts as a capacitor, able to store separated charge, and the resistance of the membrane to ion flow is high enough that small perturbations in current don't dramatically shift the potential. However, neurons are not isolated systems. They are constantly receiving synaptic inputs, as we shall explore in later chapters, and these inputs cause transient changes in membrane conductance to various ions, producing small fluctuations around the resting potential. It is worth considering what determines the specific value of the resting potential in different neurons. While negative 70 millivolts is typical, there is variation. Some neurons rest at negative 60 millivolts, others at negative 80. These differences reflect variations in the relative permeabilities to different ions, which in turn reflect differences in the types and densities of ion channels expressed in the membrane. A neuron with more potassium leak channels will rest closer to the potassium equilibrium potential. A neuron with more sodium leak channels will rest at a less negative value. The resting potential also varies across different parts of the same neuron. The dendrites, soma, and axon do not necessarily all sit at exactly the same voltage. Local differences in channel expression and in the geometry of the cell create spatial variation in the potential. However, these differences are typically small, on the order of a few millivolts, because the cytoplasm inside the cell is an excellent conductor, tending to equalize potentials across the cell's interior. Temperature affects the resting potential, though perhaps not as dramatically as one might expect. The Nernst equation includes temperature as a factor, and increasing temperature increases the thermal energy available to ions, slightly shifting equilibrium potentials. However, biological systems are buffered against temperature changes, and neurons function effectively across a range of physiologically relevant temperatures. What is more significant is that the pumps and channels themselves, being proteins, have temperature-dependent activity, and these effects can partially compensate for the purely thermodynamic changes in ionic behavior. The extracellular concentration of ions is normally tightly regulated by physiological mechanisms. Blood ion levels are maintained within narrow ranges, and the brain is further protected by the blood-brain barrier, which regulates what substances can enter the interstitial fluid surrounding neurons. However, in pathological conditions, these concentrations can change. An increase in extracellular potassium, which can occur after injury or during intense neural activity, will make the resting potential less negative, bringing the neuron closer to threshold and making it more excitable. A decrease in extracellular calcium, which affects the stability of voltage-gated sodium channels, can have similar effects. Consider the chloride ion more carefully. In many cells, chloride is passively distributed, meaning its distribution is determined by the voltage across the membrane, and it contributes little to setting that voltage. However, in neurons, chloride concentrations are actively regulated by chloride transporters that use the sodium gradient as an energy source. The potassium chloride cotransporter moves both ions in the same direction, tending to drive chloride out of the cell. As a result, in mature neurons, the chloride equilibrium potential is typically at or slightly more negative than the resting potential. This has important consequences for inhibitory neurotransmission, as we shall see later. Interestingly, the chloride equilibrium potential changes during development. In immature neurons, chloride accumulates inside cells, making the chloride equilibrium potential more positive than the resting potential. This has the surprising consequence that activation of GABA receptors, which open chloride channels, can actually depolarize immature neurons rather than hyperpolarize them, making GABA excitatory early in development rather than inhibitory as it is in adults. This developmental switch reflects changes in the expression of chloride transporters. The resting potential is not merely a passive baseline from which signals deviate. It actively influences neuronal excitability. A neuron resting at a more negative potential is further from threshold, requiring larger inputs to trigger an action potential. A neuron at a less negative potential is closer to threshold and more easily excited. Neuromodulatory systems, which we will discuss in later chapters, often work by subtly shifting the resting potential up or down, thereby adjusting how readily neurons respond to synaptic inputs. The membrane's capacitance also plays a role in how the membrane potential changes in response to current. The lipid bilayer acts as a capacitor, meaning it can store separated charge. When current is injected into a neuron, the membrane potential doesn't change instantaneously. Instead, it rises or falls with a characteristic time course determined by the membrane capacitance and resistance, typically on the order of tens of milliseconds for small neurons and longer for larger neurons with more membrane surface area. This capacitance acts as a low-pass filter, smoothing out very rapid fluctuations in current and allowing the neuron to integrate inputs over time. Different regions of the neuron can have different time constants. A dendrite with a high density of open channels has a lower resistance and thus a shorter time constant, responding more quickly to inputs, but also losing charge more rapidly to the surrounding medium. An axon, with fewer open channels at rest, has a higher resistance and a longer time constant, retaining charge more effectively and allowing signals to propagate greater distances without decaying. From a thermodynamic perspective, the neuron at rest is in a steady state rather than true equilibrium. Energy is continuously being expended to maintain concentration gradients, and these gradients are continuously being dissipated by ion leak. The system maintains a constant voltage not because it has reached equilibrium, but because the rates of charge movement into and out of the cell are exactly balanced. This is an example of a dynamic steady state common in biological systems, where constancy is achieved through continuous balancing of opposing processes rather than through the absence of processes. The existence of the resting potential is ancient, shared by all neurons across all animals, from cnidarians to humans. This universality suggests that the electrical polarization of neurons is a fundamental requirement for their function, and indeed, it is difficult to imagine how a neuron could signal without it. The resting potential provides both the energy source for action potentials, which we shall explore in the next chapter, and the baseline against which synaptic inputs are measured. It is the reference point for the electrical language of neurons. As we turn our attention next to the action potential, we will see how this carefully maintained resting state is temporarily and dramatically disrupted in a controlled manner to transmit information. The resting potential is not the end of the story, but rather the beginning, the stable platform from which neurons launch their signals. Understanding it is essential for understanding everything that follows, for without the concentration gradients and electrical polarization that characterize the resting state, neurons would have no mechanism for rapid signaling. Once an action potential is initiated at the axon hillock, it does not simply sit there, content with its brief moment of electrical reversal. Rather, it begins a journey, propagating along the length of the axon toward the terminal. This propagation is not the movement of particles flowing through a tube, like water through a pipe. Instead, it's a sequential regeneration of the electrical event, each section of membrane triggering the next in a precisely coordinated cascade. Understanding this process requires examining the local currents that spread through the cytoplasm and across the membrane, the continuous renewal of the signal, and the factors that determine how quickly this wave of depolarization travels. In unmyelinated axons, the process is straightforward, if somewhat inefficient. When the membrane at the axon hillock depolarizes and sodium rushes in, the local concentration of positive charges inside the cell increases dramatically. These positive charges don't remain stationary. They spread longitudinally through the cytoplasm, moving both forward toward the axon terminal and backward toward the soma. However, the refractory period behind the action potential prevents backward propagation. The membrane that has just fired cannot immediately fire again, creating a directional bias. The positive charges spreading forward, meanwhile, encounter membrane that is still at resting potential, approximately negative 70 millivolts. This spreading positive charge on the inside of the membrane encounters the negative charge on the inside of the adjacent resting membrane. The result is a local current flow. Positive charges move through the cytoplasm toward the more negative region, and this movement of charge begins to depolarize the adjacent patch of membrane. If this depolarization reaches threshold, approximately negative 55 millivolts, the voltage-gated sodium channels in that region open. Sodium floods in, depolarizing that section of membrane to around positive 40 millivolts, and the cycle repeats. In this way, the action potential regenerates itself continuously along the length of the axon. The speed of this continuous conduction is limited by several factors. First, the cytoplasm has electrical resistance. It is not a perfect conductor. As current flows through it, some energy is lost as heat, and the voltage change diminishes with distance. This means that the depolarization from one action potential site becomes weaker as it spreads, requiring relatively close spacing for the signal to successfully trigger the next section. Second, the membrane itself is not perfectly insulated. It's leaky. Even at rest, some ions cross through various channels, and during the spreading of local currents, capacitance effects come into play. The lipid bilayer acts as a capacitor, storing charge across its surface, which slows the rate at which voltage changes. In practical terms, continuous conduction in unmyelinated axons proceeds at roughly 0.5 to 2 meters per second, depending on the diameter of the axon. Larger diameter axons conduct faster because they have lower internal resistance. There's simply more cytoplasm for current to flow through, which reduces the resistance, much like a wide pipe allows water to flow more easily than a narrow one. However, even in relatively large unmyelinated fibers, conduction remains slow by the standards of a nervous system that often needs to react in milliseconds. The mechanism itself is remarkably reliable because each section of membrane regenerates the action potential fully. The signal does not decay over distance. An action potential that travels 1 centimeter looks identical to one that travels 1 meter. This is a crucial feature. It means that neurons can be quite long, extending from the spinal cord to the tip of a finger or toe without signal degradation. The all or nothing nature of the action potential combines with this regenerative propagation to ensure fidelity of transmission. Consider the structural details that support this process. The axon's cylindrical shape provides a continuous pathway for current flow. The cytoplasm, while resistive, is relatively homogeneous, allowing for predictable current spread. The density of voltage-gated sodium channels along the axon membrane is sufficiently high that adjacent patches can reliably trigger one another. In unmyelinated axons, these channels are distributed continuously along the membrane, with densities reaching several hundred channels per square micrometer. The exact speed of propagation can be calculated using cable theory, a mathematical framework borrowed from electrical engineering. The axon is modeled as a leaky cable with internal resistance, membrane resistance, and membrane capacitance. The length constant, a parameter describing how far a voltage change can spread before decaying, depends on the ratio of membrane resistance to internal resistance. A higher membrane resistance, better insulation, and lower internal resistance, larger diameter, both increase the length constant and thus the conduction velocity. The time constant, meanwhile, describes how quickly the membrane voltage can change, and it depends on membrane resistance and capacitance. These equations reveal why evolution has favored certain solutions. To speed up conduction in unmyelinated axons, one must either increase diameter or decrease membrane capacitance. Increasing diameter works, but only to a point. Some invertebrates, such as squid, have evolved giant axons approaching 1 millimeter in diameter to achieve rapid conduction. These axons conduct at speeds up to about 25 meters per second, which is impressive for continuous conduction. However, the space requirements are prohibitive for complex nervous systems. A human brain, constructed with such large axons, would need to be impractically enormous. Decreasing membrane capacitance is another option, and this is where myelination enters the picture. But before we discuss that, it's worth examining what happens at the molecular level during continuous conduction. As each section of membrane depolarizes, the voltage-gated sodium channels undergo their conformational changes. The activation gate opens in response to the depolarization, spreading from the adjacent active region. Sodium enters, driven by both its concentration gradient and the electrical gradient. The local membrane potential shoots up toward the sodium equilibrium potential, approximately positive 60 millivolts, though it typically peaks around positive 40 millivolts due to the opening of potassium channels. These voltage-gated potassium channels, which we've discussed previously, open in response to depolarization, but with slower kinetics than sodium channels. By the time they fully open, the sodium channels have often already begun to inactivate. The outward flow of potassium ions brings the membrane potential back down, repolarizing it, In unmyelinated axons, this repolarization must occur at each point along the axon where depolarization has occurred. The sodium potassium pump then works continuously to restore the original ion gradients, though this occurs on a slower timescale than the action potential itself. The refractory period following each action potential serves a critical function in continuous conduction. The absolute refractory period, lasting about 1 to 2 milliseconds, during which another action potential cannot be generated regardless of stimulus strength, ensures unidirectional propagation. The region of membrane immediately behind the advancing action potential cannot fire again, so the signal moves forward. The relative refractory period, during which a stronger than normal stimulus is required to trigger another action potential, limits the maximum frequency of action potentials. In most neurons, this ceiling is around 1,000 action potentials per second, though typical firing rates are much lower, in the range of 1 to 100 per second. Temperature affects conduction velocity in unmyelinated axons. Higher temperatures increase the kinetic energy of molecules, speeding up the conformational changes in ion channels and reducing the viscosity of the membrane lipids. This accelerates both depolarization and repolarization. In humans, where core body temperature is maintained near 37 degrees Celsius, this is generally not a variable factor. However, in peripheral nerves of the limbs, where temperatures can drop, conduction velocity may decrease. This is one reason why fingers feel sluggish and less responsive in cold weather. The metabolic demands of continuous conduction are not negligible. While the action potential itself doesn't require ATP, the restoration of ion gradients by the sodium-potassium pump does. Each cycle of the pump moves 3 sodium ions out and 2 potassium ions in, consuming 1 molecule of ATP. Given that millions of ions may cross the membrane during a single action potential, the energy expenditure adds up. In unmyelinated axons conducting frequently, a substantial portion of the cell's ATP budget goes toward maintaining these gradients. This is one reason why neurons are so vulnerable to interruptions in oxygen or glucose supply. Without ATP, the pump fails, gradients dissipate, and the neuron can no longer fire action potentials. Now, consider the physical journey of an action potential along a long unmyelinated axon. Imagine a sensory neuron carrying information about pain from your toe. The cell body sits in a dorsal root ganglion near the spinal cord, and its axon extends all the way to receptors in the skin of the toe, a distance that could exceed 1 meter. When those receptors are activated by, say, stubbing your toe against a piece of furniture, an action potential is generated and begins its propagation toward the spinal cord. At 1 meter per second, this signal would take a full second to arrive, which would make for a delayed and unsatisfying warning system. Another consideration is the density and distribution of voltage-gated channels. While sodium channels are essential for depolarization, potassium channels are equally important for repolarization. In unmyelinated axons, both channel types are distributed relatively uniformly along the membrane. However, different neuronal types express different subtypes of these channels, each with distinct kinetics and voltage sensitivities. This diversity allows for fine-tuning of action potential shape and conduction properties. Some neurons fire brief, spiky action potentials, others fire broader ones. These differences emerge from variations in channel composition. The continuous conduction mechanism is ancient in evolutionary terms. Invertebrates rely heavily on it, and it works well enough for their needs. Jellyfish, worms, and insects all conduct action potentials along unmyelinated axons with success, albeit at slower speeds. The evolutionary pressure to increase conduction speed without increasing axon diameter led to one of the more elegant solutions in neural architecture, the development of myelin and saltatory conduction, which we'll explore next. But it's worth noting that even in highly advanced nervous systems like our own, unmyelinated axons persist. They serve important functions, particularly in systems where speed is less critical than other factors, such as metabolic efficiency or the need for fine gradations of signal timing. Before we move forward to discuss myelination, let's take a moment to appreciate the sheer engineering involved in continuous conduction. The axon maintains a polarized membrane at rest, responds to depolarization by opening sodium channels, regenerates the signal at each successive point, repolarizes through potassium efflux, and does all of this while maintaining directional propagation and signal fidelity over distances that can span significant fractions of the body. It accomplishes this using molecular machines, ion channels, and pumps that operate with remarkable precision, switching states in microseconds and conducting ions at rates exceeding millions per second per channel. The simplicity of the mechanism belies its effectiveness. No external energy is required for the propagation itself. The electrochemical gradients provide the driving force. The neuron simply needs to maintain those gradients, which it does continuously, and the rest follows from the biophysics of charged particles moving through channels. In this sense, the action potential is a self-sustaining wave of electrochemical activity, a biological analog to a wave traveling along a rope, though with the added feature that it regenerates its own energy at each point. As we turn now to consider what happens when axons acquire an insulating sheath, we'll see how evolution took this already functional system and optimized it for speed, transforming conduction velocity from meters per second to tens of meters per second, and in the process, enabling the rapid reflexes and swift reactions that characterize higher vertebrate nervous systems. The transition from continuous to saltatory conduction represents a significant leap in neural efficiency, one that fundamentally shaped the architecture of the brain and spinal cord. The evolution of myelin represents one of the more elegant solutions to a biological engineering problem. How do you increase the speed of neural signals without dramatically increasing the size of axons? The answer, arrived at independently in vertebrates and in some invertebrates, was to wrap the axon in layers of insulating material, leaving small gaps at regular intervals. This allows the action potential to jump from gap to gap rather than propagating continuously along every millimeter of membrane. The result is a dramatic increase in conduction velocity from roughly 2 meters per second in unmyelinated fibers to as much as 120 meters per second in heavily myelinated ones. The term for this jumping conduction is saltatory, from the Latin saltare, meaning to jump or leap. Myelin is not a single structure, but rather a spiral wrapping of membrane produced by specialized glial cells. In the peripheral nervous system, these are Schwann cells, named after the German physiologist Theodor Schwann, who first described them in the 19th century. In the central nervous system, the job falls to oligodendrocytes, a name derived from Greek roots meaning few branch cells, though in practice they branch quite extensively. Both cell types perform the same essential function. They wrap their plasma membrane around the axon in concentric layers, creating a thick insulating sheath. The process begins during development. Schwann cells in the periphery align themselves along axons, with each cell taking responsibility for a single segment, typically about 1 millimeter in length. The Schwann cell extends a process that wraps around the axon multiple times, spiraling like a roll of tape. With each turn, the cytoplasm is squeezed out from between the membrane layers, leaving primarily lipid bilayers stacked tightly together. These compacted membranes, rich in cholesterol and sphingomyelin, form the myelin sheath. A single Schwann cell may wrap around an axon 20 to 150 times, depending on the fiber diameter, creating a sheath that can be 20 micrometers thick in large fibers. Oligodendrocytes in the central nervous system operate somewhat differently. Rather than each cell myelinating a single segment of one axon, a single oligodendrocyte extends processes to multiple axons, sometimes as many as 50, and myelinates a segment of each. This is a more efficient arrangement in terms of cell number, though it comes with trade-offs. If an oligodendrocyte is damaged, multiple axons lose myelination simultaneously. In contrast, damage to a Schwann cell affects only one segment of one axon. Between the myelinated segments lie the nodes of Ranvier, named after the French pathologist Louis-Antoine Ranvier, who described them in 1871. These nodes are small unmyelinated gaps, typically about 1 micrometer in length, where the axon membrane is exposed to the extracellular environment. It is at these nodes that action potentials are regenerated. The myelinated regions, called internodes, do not generate action potentials. They simply serve as insulated cables, allowing current to flow with minimal loss. The biophysics of saltatory conduction are straightforward but consequential. When an action potential occurs at one node of Ranvier, sodium rushes in and the local membrane depolarizes to around positive 40 millivolts. The positive charges spread through the axoplasm, just as in continuous conduction, but here the myelin sheath dramatically reduces the capacitance and increases the resistance of the membrane. The lipid layers prevent ions from leaking out through the internodal membrane. This means that the current spreading through the cytoplasm can travel much farther before dissipating. The result is that the depolarization from one node is sufficient to reach the next node, even though they may be 1 to 2 millimeters apart. When the spreading positive charge reaches the next node, it depolarizes that node's membrane to threshold, triggering the opening of voltage-gated sodium channels concentrated there. The action potential is regenerated, and the process repeats. The signal effectively jumps from node to node, bypassing the myelinated internodes. This arrangement increases conduction velocity for several reasons. First, the action potential need only be regenerated at the nodes, rather than continuously along the entire axon. This reduces the time required for the signal to travel a given distance. Second, the myelin reduces membrane capacitance. Capacitance is inversely proportional to the distance between the two conducting surfaces, in this case, the inner and outer faces of the membrane. By adding many layers of membrane, myelin increases this distance, thereby decreasing capacitance. Lower capacitance means the membrane voltage can change more rapidly, speeding up the depolarization at each node. Third, the high membrane resistance of the myelin sheath prevents current from leaking out during the internodal conduction. In cable theory terms, myelin increases the length constant, allowing current to spread farther before decaying. The combination of reduced capacitance and increased resistance dramatically improves the efficiency of current spread, which translates directly into faster conduction. The relationship between axon diameter, myelin thickness, and conduction velocity is not arbitrary. For optimal conduction speed, the ratio of axon diameter to total fiber diameter, including myelin, should be around zero point 0.6 to zero point 0.7. This ratio, known as the G ratio, balances the need for sufficient axoplasm to carry current with the need for adequate insulation. Too little myelin, and the benefits are reduced. Too much, and the axon diameter is unnecessarily constrained, increasing internal resistance. The voltage-gated sodium channels at the nodes of Ranvier are present in extraordinarily high densities, often exceeding 1,000 channels per square micrometer. This is roughly 10 times the density found in unmyelinated axons. The high density ensures that even a weakened depolarization arriving from the previous node can trigger a full-fledged action potential. The nodes also contain high densities of voltage-gated potassium channels, which handle repolarization. Interestingly, the potassium channels at the nodes often include fast-activating subtypes that speed up the return to resting potential, further increasing the maximum firing frequency. The internodal regions, meanwhile, have relatively few voltage-gated channels. They don't need them. The myelin prevents significant ion flux across the membrane, and the propagation through these regions is purely passive. It's a bit like the difference between a relay race, where runners hand off a baton, and a single runner covering the entire distance. In continuous conduction, the signal is a single runner regenerating itself at every step. In saltatory conduction, the signal is passed between relay stations, with passive coasting in between. One might wonder why the nodes are necessary at all. Why not wrap the entire axon in myelin and allow a single action potential to propagate all the way to the terminal through passive spread alone? The answer lies in distance and attenuation. Even with perfect insulation, current spreading through the cytoplasm encounters resistance. The voltage change diminishes with distance according to an exponential decay. For most axons, which can extend for centimeters or even meters, a single action potential would decay to subthreshold levels long before reaching the end. The nodes of Ranvier solve this by periodically regenerating the signal, ensuring that it arrives at the axon terminal with full amplitude. The spacing of the nodes is not arbitrary. It's optimized for the diameter of the axon. Larger diameter axons have longer internodes, sometimes exceeding 2 millimeters, because their lower internal resistance allows current to spread farther. Smaller diameter axons have shorter internodes, often less than 1 millimeter. This relationship ensures that each axon conducts as rapidly as possible given its diameter. The metabolic advantages of saltatory conduction are substantial because the action potential is only regenerated at the nodes. Fewer ions cross the membrane overall compared to continuous conduction. This means less work for the sodium-potassium pump and thus lower ATP consumption per action potential. Estimates suggest that myelinated axons require only about 1/100th the energy of unmyelinated axons to conduct the same signal over the same distance. This efficiency becomes critical in the brain, where billions of neurons fire continuously and energy budgets are tight. The development of myelin during growth is a protracted process. In humans, myelination begins in utero, but continues well into adolescence, and in some regions into the third decade of life. The process follows a general pattern. Motor pathways myelinate before sensory pathways, and caudal regions myelinate before rostral ones. The last regions to fully myelinate are the prefrontal cortex and other association areas involved in higher cognitive functions. This prolonged myelination correlates with the development of various cognitive and motor abilities. Infants, with incompletely myelinated motor tracts, exhibit jerky, uncoordinated movements. As myelination progresses, movements become smoother and more precise. White matter in the brain and spinal cord derives its color from myelin. The lipid-rich sheaths are pale, almost white, especially when compared to gray matter, which is densely packed with neuronal cell bodies, dendrites, and unmyelinated axons. The optic nerve, for instance, appears white because it consists almost entirely of myelinated axons carrying visual information from the retina to the brain. In fresh tissue, the distinction is striking. In preserved specimens, the contrast is more muted, but still evident. The molecular composition of myelin is worth examining. By dry weight, myelin is about 70 to 85% lipid and 15 to 30% protein. The high lipid content is what makes it an effective insulator. Among the lipids, cholesterol is particularly abundant, comprising about 25% of all myelin lipids. Phospholipids, including phosphatidylcholine and phosphatidylethanolamine, make up much of the rest. The protein component includes myelin basic protein, MBP, and proteolipid protein, PLP, in the central nervous system, and myelin protein 0, MPZ, in the peripheral nervous system. These proteins help stabilize the compact myelin structure and play roles in myelin formation and maintenance. Mutations in myelin proteins can cause severe neurological diseases. Charcot-Marie-Tooth disease, a group of inherited neuropathies, often results from mutations in genes encoding myelin proteins or proteins involved in Schwann cell function. Affected individuals experience progressive weakness and atrophy of distal muscles, along with sensory loss, as their peripheral nerves lose myelin and conduction slows. Pelizaeus-Merzbacher disease, caused by mutations in the PLP gene, leads to defective myelination in the central nervous system, resulting in severe developmental delays, spasticity, and ataxia. The autoimmune destruction of myelin is the hallmark of multiple sclerosis, a disease we'll discuss more in a later chapter. For now, it's sufficient to note that the loss of myelin has predictable consequences. Conduction velocity slows dramatically. In some cases, conduction fails altogether as the passive current spread between nodes becomes insufficient to trigger action potentials. The clinical manifestations depend on which axons are affected. Demyelination in the optic nerve causes vision problems, in motor tracts, weakness and incoordination, in sensory tracts, numbness and tingling. The nodes of Ranvier are not simply gaps, they are highly organized structures. The axon membrane at the node is anchored by cytoskeletal proteins that cluster the voltage-gated sodium channels. Flanking the node are perinodal regions where the myelin loops attach to the axon membrane through specialized junctions. These junctions prevent the lateral diffusion of channels out of the nodal region and maintain the distinct molecular organization of the node. Farther from the node are juxtaparanodal regions, which contain voltage-gated potassium channels that are sequestered away from the node itself by the paranodal junctions. This precise molecular architecture is maintained dynamically. If myelin is damaged, the organization of the node can be disrupted. Sodium channels may diffuse away, reducing the node's excitability. Potassium channels may invade the nodal region, causing excessive repolarization that suppresses action potential generation. Remyelination, when it occurs, attempts to restore this organization, though it's often imperfect. Schwann cells and oligodendrocytes do more than just provide insulation, they also support the axons metabolically. The glial cells provide lactate, which axons can use as an energy substrate. They regulate the local ionic environment, clearing excess potassium that accumulates in the narrow extracellular space during action potentials. They secrete neurotrophic factors that promote axonal health and survival. In this sense, myelination is not just a structural modification, but a functional partnership between neuron and glia. Saltatory conduction also enables neurons to fire at higher frequencies. The brief duration of the action potential at each node, combined with the rapid passive spread through the internodes, means that a second action potential can follow closely behind the first. The refractory period still limits maximum frequency, but the overall conduction time is so much shorter that neurons can sustain higher firing rates without signals colliding or interfering with one another. In the peripheral nervous system, different sensory modalities rely on different degrees of myelination. Proprioceptors, which signal the position and movement of limbs, send their information along heavily myelinated fibers, conducting at 70 to 120 meters per second. This speed is essential for fine motor control and balance. Touch receptors use moderately myelinated fibers, conducting at 30 to 70 meters per second. Pain and temperature, meanwhile, use either thinly myelinated or unmyelinated fibers, conducting at 0.5 to 30 meters per second. The slower conduction of pain signals explains the delay between injury and the sensation of pain, a delay that can sometimes seem merciful, other times merely puzzling. In summary, saltatory conduction transforms the neuron into a high-speed communication line. By insulating the axon with myelin and concentrating the regenerative machinery at the nodes of Ranvier, the nervous system achieves conduction velocities that would require impossibly large axon diameters if continuous conduction were the only option. The efficiency, both in terms of speed and energy, is remarkable. And yet, for all this sophistication, the action potential must still ultimately reach its destination, the axon terminal, where the electrical signal will be converted into a chemical one at the synapse. The action potential, having traveled the length of the axon through continuous or saltatory conduction, now arrives at the axon terminal. This is where the electrical nature of the signal undergoes a transformation. For most synapses in the nervous system, the signal cannot simply jump from one neuron to the next. There is a gap, a physical discontinuity measuring roughly 20 to 40 nanometers across, called the synaptic cleft. This tiny space, barely visible under a light microscope, requires the signal to change from electrical to chemical and then back to electrical on the other side. It is an arrangement that seems inefficient at first glance, but it provides the nervous system with remarkable flexibility, modularity, and computational power. The synapse, in its most common form, consists of three components: the presynaptic terminal, the synaptic cleft, and the postsynaptic membrane. The presynaptic terminal is the enlarged ending of the axon, a bulb-shaped or button-like structure packed with specialized machinery. The postsynaptic membrane is usually part of a dendrite or cell body of another neuron, though it can also be a muscle cell or gland cell. The cleft is the narrow extracellular space separating the two. The entire structure is a specialized contact point, and the human brain contains an estimated 100 trillion of them. Let's begin with the presynaptic terminal. This structure is, in many ways, a factory dedicated to the production, storage, and release of neurotransmitters. The cytoplasm here is crowded with synaptic vesicles, small membrane-bound spheres, typically 40 to 50 nanometers in diameter, each filled with thousands of neurotransmitter molecules. These vesicles are manufactured in the cell body and transported down the axon via motor proteins that walk along microtubules, a journey that can take hours or even days depending on the length of the axon. The presynaptic membrane itself is studded with various proteins. Most notably, there are voltage gated calcium channels. These channels are closed at resting membrane potential, but when an action potential arrives at the terminal, the depolarization opens them. Calcium ions, which are far more concentrated outside the cell, around 2 millimolar in the extracellular fluid versus about 0.0001 millimolar in the cytoplasm, rush in down their steep electrochemical gradient. This influx of calcium is the critical trigger for neurotransmitter release. The concentration of calcium in the presynaptic terminal rises rapidly, often increasing 100-fold within a fraction of a millisecond. This is not a global change, but rather a localized spike near the inner mouth of the calcium channels. The synaptic vesicles are not distributed randomly. They are organized into pools. Some are docked at the presynaptic membrane at specialized sites called active zones. These docked vesicles are primed and ready to fuse with the membrane. Others are held in reserve, tethered to the cytoskeleton nearby, and still others are part of a recycling pool, vesicles that have recently been retrieved from the membrane after a previous release event. The architecture of the active zone is a marvel of molecular organization. The presynaptic membrane here is thickened by a dense meshwork of proteins, including scaffolding proteins that anchor the calcium channels close to the docked vesicles. This proximity is crucial. The calcium signal is brief and highly localized. The vesicles must be positioned within a few tens of nanometers of the calcium channels to sense the increase in calcium concentration. If they were farther away, the calcium would diffuse and be buffered by calcium-binding proteins before it could trigger fusion. Now let's turn to the synaptic cleft. This space, though tiny, is not empty. It is filled with extracellular fluid, a salt solution rich in sodium and chloride, with lower concentrations of potassium and calcium. The cleft also contains a meshwork of extracellular matrix proteins, glycoproteins that bridge the presynaptic and postsynaptic membranes, holding them at a fixed distance. These adhesion molecules, such as neurexins on the presynaptic side and neurologens on the postsynaptic side, bind to each other across the cleft. They not only provide structural stability, but also play roles in synapse formation and maturation during development. The width of the cleft is not arbitrary. 20 to 40 nanometers is narrow enough that neurotransmitters can diffuse across it rapidly, typically in less than a millisecond, but wide enough that the two membranes remain electrically isolated. This prevents the action potential from simply crossing over by passive current flow, which could happen if the membranes were closer. The cleft is also wide enough to accommodate enzymes that degrade neurotransmitters, ensuring that signals are terminated promptly. On the postsynaptic side, the membrane is densely packed with receptors, proteins that bind neurotransmitters and initiate a response in the postsynaptic cell. These receptors are not scattered randomly. They are concentrated in the region of the membrane directly opposite the presynaptic active zone. This organization, known as the postsynaptic density in central nervous system synapses, is a thick, electron-dense structure visible in electron micrographs. It contains not only receptors, but also scaffolding proteins, signaling enzymes, and cytoskeletal elements. The postsynaptic density anchors the receptors in place and provides a platform for signal transduction pathways that can modify the strength of the synapse. The receptors themselves come in two broad categories, which we'll explore in more detail later: ionotropic receptors, which are ligand gated ion channels that open when neurotransmitter binds, allowing ions to flow across the membrane, and metabotropic receptors, which are coupled to intracellular signaling cascades via G proteins and produce slower, longer lasting effects. For now, what matters is that these receptors are the target of the neurotransmitters released from the presynaptic terminal. The geometry of the synapse varies depending on its location and function. Exodendritic synapses, where an axon terminal contacts a dendrite, are the most common. Axosomatic synapses, where the terminal contacts the cell body, are also frequent, particularly for inhibitory synapses. Axoaxonic synapses, where one axon terminal contacts another axon terminal, are less common but play important roles in modulating neurotransmitter release, a process called presynaptic inhibition or facilitation. There are even dendrodendritic synapses, where dendrites of different neurons make synaptic contacts with each other, though these are relatively rare and found primarily in specialized regions like the olfactory bulb. The size of the synapse also varies. Small synapses, with active zones of less than 0.1 square micrometers, may contain only a handful of docked vesicles and release neurotransmitter probabilistically. A single action potential may or may not trigger release at such synapses. Large synapses, with active zones exceeding 1 square micrometer, can have dozens or even hundreds of docked vesicles and release neurotransmitter reliably with each action potential. The size and number of synapses a neuron makes reflects its function. Neurons involved in precise timing often make large, reliable synapses. Neurons involved in integrating many inputs may make numerous small synapses, each contributing a weak signal that must be summed with others. The synaptic cleft is not merely a passive gap, it is an active participant in synaptic transmission. For one, the cleft contains enzymes that regulate neurotransmitter concentration. Acetylcholinesterase, for instance, is anchored in the cleft at neuromuscular junctions and at certain central synapses, where it rapidly breaks down acetylcholine into acetate and choline. This ensures that acetylcholine acts only briefly before being inactivated. Similarly, peptidases in the cleft can cleave neuropeptides, terminating their action. The cleft also contains molecules that modulate synaptic function. Neuromodulators, which we'll discuss in a later chapter, can be released into the cleft and affect both presynaptic and postsynaptic function without directly triggering fast synaptic responses. The extracellular matrix proteins in the cleft can be modified by enzymatic activity, and these modifications can alter synapse structure and function, contributing to synaptic plasticity. The ionic composition of the cleft can change during synaptic activity. When neurotransmitters are released and ions flow through postsynaptic receptors, the local concentrations of sodium, potassium, and calcium in the cleft can shift. These changes are usually small and transient, but in cases of intense activity, they can become significant enough to affect synaptic transmission. For example, accumulation of potassium in the cleft can depolarize both the presynaptic terminal and the postsynaptic membrane, enhancing excitability, but also potentially leading to overexcitation. One might wonder why chemical synapses evolved in the first place. Electrical synapses, which we've not yet discussed in detail, do exist. They are formed by gap junctions, channels that directly connect the cytoplasm of two cells, allowing ions and small molecules to pass freely. Electrical synapses are fast, bidirectional, and metabolically cheap. They're used in situations where speed and synchrony are paramount, such as in cardiac muscle and in certain neural circuits that require tightly coordinated activity. The electron microscope has been instrumental in revealing the structure of synapses, fixed and stained brain tissue, when viewed at magnifications of 50,000 times or more, shows the presynaptic terminal filled with circular profiles, the synaptic vesicles, the narrow cleft, and the thickened postsynaptic density. Different types of synapses can be distinguished by their morphology. Asymmetric synapses with a prominent postsynaptic density are typically excitatory. Symmetric synapses with less pronounced densities on both sides are typically inhibitory. These distinctions were first made in the 1960 seconds and remain a cornerstone of synaptic classification. More recently, advanced imaging techniques have allowed scientists to observe synapses in living tissue. Fluorescent proteins that label synaptic vesicles or receptors can be imaged with confocal or two-photon microscopy, revealing the dynamics of synapse formation, modification, and elimination in real time. These studies have shown that synapses are far more dynamic than once thought, with structural changes occurring over minutes to hours in response to activity. The presynaptic terminal contains not only synaptic vesicles, but also mitochondria, which are essential for meeting the energy demands of neurotransmitter synthesis, vesicle trafficking, and calcium buffering. Mitochondria are often found near active zones, positioned to supply ATP where it's most needed. The endoplasmic reticulum also extends into some terminals, providing calcium stores and contributing to calcium signaling. The vesicles themselves are marvels of cellular engineering. Their membranes contain specific proteins that mediate vesicle trafficking, docking, and fusion. Synapsins, for example, are proteins that tether vesicles to the cytoskeleton, keeping them in reserve until needed. Rab proteins regulate vesicle targeting and docking, and the snare proteins, which we'll discuss in the next chapter, mediate the actual fusion of the vesicle membrane with the presynaptic membrane. Each vesicle contains not just neurotransmitters, but also protons. The interior of a synaptic vesicle is acidic, with a pH around 5.5, maintained by a proton pump in the vesicle membrane. This acidity is necessary for loading neurotransmitters into the vesicle. Neurotransmitter transporters in the vesicle membrane use the proton gradient as an energy source, exchanging protons for neurotransmitter molecules. When the vesicle fuses with the presynaptic membrane and releases its contents, the protons are also released into the cleft, causing a transient acidification that can modulate receptor function. The postsynaptic membrane, meanwhile, is not simply a passive receiver. It actively regulates the number and type of receptors present, inserting new receptors from internal stores, or removing existing ones through endocytosis. This receptor trafficking is a key mechanism of synaptic plasticity. If a synapse is frequently active, the postsynaptic cell may insert more receptors, strengthening the synapse. If a synapse is rarely active, receptors may be removed, weakening it. The synaptic cleft, then, is far more than a gap to be crossed. It is a structured, dynamic space where electrical signals are translated into chemical signals with precision and flexibility. The architecture of the synapse, the organization of the presynaptic terminal, the molecular composition of the cleft, and the receptor-studded postsynaptic membrane all contribute to the remarkable feat of neurotransmission. And it is to the process of neurotransmitter release, the moment when vesicles fuse with the membrane and spill their contents into the cleft, that we now turn our attention. The arrival of an action potential at the presynaptic terminal sets in motion a cascade of events that culminates in the release of neurotransmitter molecules into the synaptic cleft. This process, known as exocytosis, is one of the fastest and most tightly regulated membrane fusion events in biology. From the opening of calcium channels to the diffusion of neurotransmitter across the cleft, the entire sequence unfolds in less than a millisecond. The machinery that accomplishes this is a collection of proteins that have been conserved across evolution, appearing not only in neurons, but in other cells that secrete substances, from insulin-releasing pancreatic cells to histamine-releasing mast cells. Let us begin with the action potential's arrival. The depolarization sweeps into the terminal, and the voltage-gated calcium channels embedded in the presynaptic membrane respond by opening. These channels are not randomly distributed. They are clustered at the active zones, the specialized regions where synaptic vesicles dock and fuse. The channels themselves are complex proteins, typically of the N-type or P divided by Q-type variety in most central nervous system synapses, with multiple subunits forming a pore through which calcium ions can pass. When the channels open, calcium rushes in. The driving force is substantial. The concentration gradient, 2 millimolar outside versus 0.0001 millimolar inside, combined with the electrical gradient, the interior is still relatively negative after the action potential passes, propels calcium ions through the pore at a rate of millions of ions per second per channel. Within a few hundred microseconds, the calcium concentration near the inner mouth of the channels rises from 0.0001 millimolar to 10 or even 100 millimolar. This is a transient, highly localized spike, often called a calcium microdomain or nanodomain, extending only a few tens of nanometers from the channel. The synaptic vesicles docked at the active zones are positioned within this calcium microdomain. They are not simply resting against the membrane. They are primed, held in a state of readiness by a complex of proteins that have brought the vesicle membrane and the presynaptic membrane into close apposition. This priming process involves partial assembly of the snare complex, the molecular machine that will ultimately pull the two membranes together and catalyze their fusion. Snare proteins are the central players in exocytosis. The name stands for soluble N-ethylmaleimide sensitive factor attachment protein receptor, a phrase that reveals the convoluted history of their discovery rather than their function. There are three key snare proteins at the synapse: synaptobrevin, also called VAMP, which is embedded in the vesicle membrane, and syntaxin and SNAP25, which are anchored in the presynaptic membrane. Before calcium arrives, these proteins have already begun to interact, forming a loose complex. When calcium binds to the calcium sensor protein synaptotagmin, which is embedded in the vesicle membrane with its calcium binding domains projecting into the cytoplasm, it undergoes a conformational change. Synaptotagmin has two C2 domains, each capable of binding multiple calcium ions. When calcium binds, these domains insert into the lipid bilayer of the presynaptic membrane, and synaptotagmin also interacts with the snare complex and with membrane phospholipids. The exact sequence of events is still debated, but the outcome is clear. Synaptotagmin triggers the completion of snare complex assembly. The snare proteins zipper together, forming a tight bundle of four alpha helices, one from synaptobrevin, one from syntaxin, and two from SNAP2-5. As this zippering proceeds from the ends of the helices nearest the membranes toward the membrane-spanning domains, it pulls the vesicle membrane and the presynaptic membrane into direct contact. The energy released by snare complex formation is sufficient to overcome the repulsive forces between the two lipid bilayers and to induce membrane fusion. Fusion begins with the formation of a fusion pore, a narrow channel connecting the interior of the vesicle with the extracellular space of the synaptic cleft. The pore opens rapidly, expanding within microseconds to allow the neurotransmitter molecules inside the vesicle to escape. A single vesicle may contain several thousand neurotransmitter molecules. Acetylcholine vesicles, for example, contain about 5,000 molecules, while glutamate vesicles may contain 2,000 to 4,000. These molecules, along with ATP, protons, and other co-stored substances, spill out into the cleft. The fusion event is not always complete. Sometimes the fusion pore opens briefly and then closes again, a process called kiss and run. In this mode, only a fraction of the vesicle's contents are released before the vesicle detaches from the membrane and is retrieved intact. Other times, the vesicle fully collapses into the presynaptic membrane, a process called full fusion, and must then be retrieved and reformed through endocytosis. The relative prevalence of these two modes varies depending on the synapse and the pattern of activity. Once fusion has occurred and neurotransmitter has been released, the snare complex must be disassembled to allow the snare proteins to participate in another round of fusion. This disassembly is accomplished by NSF, N-ethylmalamide sensitive factor, an ATPase that uses the energy from ATP hydrolysis to pull the snare bundle apart. NSF, with the help of adapter proteins called SNAPs, soluble NSF attachment proteins, not to be confused with SNAP2-5, binds to the assembled snare complex and forcibly unwinds it. This resets the snares, allowing them to be reused. The speed of neurotransmitter release is remarkable, from the opening of calcium channels to the opening of the fusion pore. The delay is typically 200 to 500 microseconds. This is one of the fastest biochemical processes in the cell, a testament to the elegance of the molecular machinery and the importance of rapid synaptic transmission. For comparison, most enzymatic reactions take milliseconds, and cellular processes like protein synthesis take minutes. The number of vesicles that fuse in response to a single action potential varies. At some synapses, such as the neuromuscular junction, where reliability is paramount, several hundred vesicles may fuse. At many central nervous system synapses, the number is much smaller, 1 to 5 vesicles on average, and sometimes none at all. This probabilistic release is a feature, not a bug. It allows synapses to operate over a range of strengths and to be modulated by various factors. The probability of release depends on several factors. The first is the probability that a calcium channel near a docked vesicle will open when the action potential arrives. Not all channels open with every action potential. They open stochastically. The second is the calcium sensitivity of the release machinery. At some synapses, a small increase in calcium is sufficient to trigger fusion. At others, a larger increase is required. The third is the number of docked primed vesicles available at the active zone. If all the readily releasable vesicles have been used up by previous action potentials, subsequent action potentials may fail to trigger release until vesicles are replenished. This brings us to the concept of vesicle pools. The vesicles in the presynaptic terminal are organized into functional groups. The readily releasable pool consists of vesicles that are docked and primed at the active zone, ready to fuse with the next action potential. This pool is small, typically containing 5 to 20 vesicles per active zone. The recycling pool consists of vesicles that have been recently retrieved from the membrane and are being refilled with neurotransmitter. The reserve pool consists of vesicles tethered to the cytoskeleton farther from the active zone. These reserve vesicles can be mobilized during sustained activity, but they require additional steps to be recruited to the active zone. Vesicle recycling is essential for maintaining neurotransmitter release during prolonged activity. After a vesicle fuses with the membrane, its proteins must be retrieved, the vesicle must be reformed, and it must be refilled with neurotransmitter. This can happen locally at the terminal through several pathways. In the fastest pathway, clathrin-mediated endocytosis, vesicle proteins are recognized by adapter proteins that recruit clathrin, a protein that forms a lattice around the budding vesicle. Dynamin, a GTPase, pinches off the vesicle from the membrane. The newly formed vesicle is uncoated, refilled with neurotransmitter by vesicular transporters, and returned to the pool of available vesicles. This entire cycle can take as little as 10 to 20 seconds. There are also slower pathways involving bulk endocytosis, where large sections of membrane are retrieved and then subdivided into vesicles, and kiss and run, which bypasses full fusion altogether. The choice of pathway depends on the pattern and intensity of activity. During intense stimulation, bulk endocytosis may predominate because it can retrieve membrane more quickly, even if the subsequent processing is slower. The refilling of vesicles with neurotransmitter is accomplished by vesicular transporters, proteins embedded in the vesicle membrane. These transporters are specific to particular neurotransmitters. Vesicular glutamate transporters, VGLUTs, load glutamate into vesicles. Vesicular GABA transporters, VGAT, load GABA and glycine. Vesicular acetylcholine transporters, VACT, load acetylcholine, and vesicular monoamine transporters, VMATs, load monoamines like dopamine, serotonin, and norepinephrine. These transporters use the proton gradient across the vesicle membrane as an energy source. As mentioned earlier, vesicles are acidic inside, maintained by a vacuolar H plus ATPase that pumps protons into the vesicle at the expense of ATP. The vesicular transporters exploit this gradient, exchanging protons for neurotransmitter molecules. This coupling ensures that vesicles can accumulate neurotransmitter against its concentration gradient, reaching concentrations inside the vesicle that far exceed those in the cytoplasm. The regulation of neurotransmitter release is complex and multifaceted. It can be modulated by changing the amount of calcium that enters the terminal, by altering the calcium sensitivity of the release machinery, or by changing the size of the readily releasable pool. Presynaptic receptors, including autoreceptors that respond to the neuron's own neurotransmitter, can inhibit or enhance release. For example, many axon terminals have autoreceptors that, when activated by neurotransmitter in the cleft, trigger intracellular signaling cascades that reduce calcium influx or decrease the probability of vesicle fusion. This provides negative feedback, preventing excessive release. Conversely, other presynaptic receptors can enhance release. At some synapses, activation of metabotropic glutamate receptors or adenosine receptors on the presynaptic terminal increases calcium influx or enhances vesicle priming. This allows one neuron to modulate the release from another, a process called heterosynaptic modulation. The molecular choreography of exocytosis has been studied in exquisite detail using a combination of techniques. Electrophysiology can measure the timing of release by recording the postsynaptic currents that result, fluorescent dyes that bind to synaptic vesicles or that are loaded into vesicles and released upon fusion allow visualization of release events in real time. Electron microscopy reveals the ultrastructure of the presynaptic terminal and the vesicles. Biochemical and genetic approaches have identified the proteins involved and dissected their functions. One of the key breakthroughs came from studying the effects of bacterial toxins. Botulinum toxin and tetanus toxin, produced by Clostridium bacteria, are among the most potent toxins known. They are proteases that specifically cleave snare proteins, preventing vesicle fusion and blocking neurotransmitter release. Botulinum toxin, which cleaves SNAP25 or syntaxin, depending on the serotype, blocks release at peripheral neuromuscular junctions, causing paralysis. Tetanus toxin, which cleaves synaptobrevin, blocks inhibitory neurotransmitter release in the spinal cord, causing uncontrolled muscle contractions. The specificity of these toxins confirmed the essential role of snare proteins in exocytosis and provided tools for studying the process. Another important discovery was the identification of synaptotagmin as the calcium sensor. Mutations in synaptotagmin that disrupt calcium binding abolish fast, synchronous neurotransmitter release while leaving slower, asynchronous release intact. This showed that synaptotagmin is specifically responsible for coupling calcium influx to rapid vesicle fusion. The stoichiometry of release is also noteworthy. While a single vesicle releases thousands of neurotransmitter molecules, the effect on the postsynaptic cell depends on how many vesicles fuse and how many receptors are available. At some synapses, the release of a single vesicle is sufficient to trigger a detectable postsynaptic response, a quantum of neurotransmission. At other synapses, multiple vesicles must fuse simultaneously for the postsynaptic cell to reach threshold for firing an action potential. The quantal nature of neurotransmitter release was first demonstrated at the neuromuscular junction by Bernard Katz and colleagues in the 1,950 seconds. They showed that even at rest, small spontaneous depolarizations of the muscle membrane occurred, each corresponding to the release of a single vesicle. These miniature end plate potentials, or minis, provided direct evidence that neurotransmitter is released in discrete packets. When an action potential arrived, the response was a larger end plate potential composed of many minis summed together. This quantal hypothesis has since been confirmed at synapses throughout the nervous system. Neurotransmitter release is also sensitive to the residual calcium that remains in the terminal after an action potential. If action potentials arrive in rapid succession, the calcium from each one adds to the residual calcium left by the previous ones. This summation of calcium signals can lead to facilitation, where the second action potential causes more release than the first, or to potentiation, where release is enhanced for several seconds after a burst of activity. These forms of short-term synaptic plasticity contribute to the computational properties of neural circuits. In summary, neurotransmitter release is a precisely orchestrated process involving vesicle docking, priming, calcium sensing, snare-mediated fusion, and vesicle recycling. It is fast, efficient, and highly regulated, allowing neurons to communicate with millisecond precision while remaining adaptable to changes in activity. The release of neurotransmitter into the synaptic cleft is the culmination of the electrical signal that began as an action potential. Now, these chemical messengers must cross the cleft and bind to receptors on the postsynaptic membrane, translating the chemical signal back into an electrical one. The neurotransmitter molecules, having been released into the synaptic cleft, now diffuse across that narrow gap toward the postsynaptic membrane. Diffusion, at such short distances and high concentrations, is rapid. Within 10 to 100 microseconds, the first molecules reach the postsynaptic receptors. These receptors are proteins embedded in the membrane, each folded into a specific three-dimensional shape that allows it to bind a particular neurotransmitter. The binding event is often described using the lock and key metaphor, though this understates the dynamic nature of the interaction. Receptors are not rigid locks. They are flexible molecules that undergo conformational changes upon binding, and these changes are what transduce the chemical signal into a cellular response. Receptors fall into two broad categories, each operating on different timescales and through different mechanisms. Ionotropic receptors are ligand-gated ion channels. They are receptors and channels in one. When neurotransmitter binds, the channel opens, allowing ions to flow across the membrane. The response is fast, beginning within a millisecond of binding. Metabotropic receptors, by contrast, do not form channels themselves. Instead, they are coupled to intracellular signaling pathways via G proteins. When neurotransmitter binds, the receptor activates a G protein, which in turn activates enzymes or modulates ion channels indirectly. The response is slower, taking tens of milliseconds to seconds, but it can be longer lasting and more diverse in its effects. Let us begin with ionotropic receptors. These are large multi-subunit proteins. A typical ionotropic receptor consists of 4 or 5 subunits arranged around a central pore. Each subunit contributes to the walls of the pore and to the binding sites for the neurotransmitter. The nicotinic acetylcholine receptor, one of the first to be characterized in detail, serves as a useful example. The nicotinic receptor is found at the neuromuscular junction and in certain neurons. It consists of 5 subunits, 2 alpha, 1 beta, 1 gamma, or epsilon in adult muscle, and 1 delta arranged in a ring. Each alpha subunit has a binding site for acetylcholine, and both sites must be occupied for the channel to open efficiently. When acetylcholine molecules diffuse across the cleft and bind to these sites, they stabilize a conformation of the protein in which the pore is open. The pore, which runs through the center of the ring of subunits, is lined with amino acids that make it permeable to cations sodium, potassium, and to a lesser extent calcium, but not to anions. Other ionotropic receptors follow similar principles, but differ in their specifics. Glutamate receptors, the major excitatory receptors in the brain, come in 3 main types, named after synthetic agonists that selectively activate them: AMPA, kainate, and NMDA receptors. AMPA receptors are the workhorses of fast excitatory transmission. They consist of 4 subunits, each with a binding site for glutamate. When glutamate binds, the channel opens, allowing sodium and potassium to flow. The result is a rapid EPSP. NMDA receptors are more complex. They also bind glutamate, but they have several unique features. First, they require the binding of a coagonist, either glycine or D-serine, to open. Second, at resting membrane potential, the NMDA receptor channel is blocked by magnesium ions that sit in the pore. The receptor will only conduct ions if the membrane is already somewhat depolarized, which displaces the magnesium. Third, when the channel does open, it is permeable not only to sodium and potassium, but also to calcium. The calcium influx through NMDA receptors is particularly important for triggering intracellular signaling cascades that underlie synaptic plasticity. This voltage-dependent magnesium block makes the NMDA receptor a coincidence detector. It conducts significantly only when two conditions are met. Glutamate is present in the cleft, indicating presynaptic activity, and the postsynaptic membrane is depolarized, indicating postsynaptic activity. This property allows NMDA receptors to detect correlated activity between the presynaptic and postsynaptic neurons, a feature that is central to Hebbian learning mechanisms. Inhibitory ionotropic receptors operate on the same basic principles but with different ion selectivities. GABA-A receptors, the major inhibitory receptors in the brain, bind the neurotransmitter GABA gamma aminobutyric acid. Like the nicotinic receptor, they consist of 5 subunits arranged around a central pore. However, the pore is selective for chloride ions. When GABA binds and the channel opens, chloride flows into the cell or out, depending on the chloride gradient, but in most mature neurons, it flows in. The influx of negative charge hyperpolarizes the membrane, moving it away from the threshold for firing and action potential. This hyperpolarization is an inhibitory postsynaptic potential, or IPSP. Glycine receptors, another class of inhibitory receptors, are found primarily in the spinal cord and brainstem. They also conduct chloride and produce IPSPs when glycine binds. Structurally, they resemble GABA-A receptors, and both belong to the same superfamily of ligand-gated ion channels, known as the cis loop receptors because of a conserved cysteine loop in their structure. The kinetics of these receptors, how quickly they open, how long they stay open, and how quickly they close, vary and contribute to the temporal properties of synaptic transmission. AMPA receptors, for instance, open and close rapidly within a few milliseconds. This produces a brief EPSP that tracks the time course of glutamate in the cleft. NMDA receptors, by contrast, have slower kinetics. Once opened, they remain open for tens to hundreds of milliseconds, producing a prolonged EPSP. GABA-A receptors have intermediate kinetics, with openings lasting several milliseconds. The number of receptors at a synapse is not fixed. It can be regulated by the postsynaptic cell in response to activity, a process called receptor trafficking. Receptors are inserted into the membrane from intracellular stores or removed by endocytosis. If a synapse is frequently active, the postsynaptic cell may insert more AMPA receptors, increasing the synapse's strength. If a synapse is inactive, AMPA receptors may be removed, weakening it. This receptor trafficking is a key mechanism of synaptic plasticity, which we'll explore in a later chapter. The diversity of receptor subunits adds another layer of complexity. Each type of ionotropic receptor can be assembled from multiple subunit types, and the specific combination of subunits affects the receptor's properties. AMPA receptors, for example, can contain GLUA1, GLUA2, GLUA3, or GLUA4 subunits in various combinations. Receptors containing GLUA2 are calcium impermeable, while those lacking it are calcium permeable. GABA-A receptors can be assembled from alpha, beta, gamma, delta, epsilon, pi, theta, and rho subunits, with 19 different genes in total. The composition of a GABA-A receptor affects its sensitivity to GABA, its kinetics, and its sensitivity to drugs like benzodiazepines and barbiturates. This subunit diversity means that synapses can be tuned to different functional requirements. Fast synapses use receptors with rapid kinetics. Synapses involved in plasticity may use receptors with slower kinetics and calcium permeability. Synapses that need to be sensitive to low concentrations of neurotransmitter may use receptor subtypes with high affinity for the transmitter. Now let us turn to metabotropic receptors. These are 7 transmembrane proteins, meaning they snake through the membrane 7 times. They belong to the G protein coupled receptor, GPCR, superfamily, one of the largest and most diverse protein families in the genome. Thousands of different GPCRs exist, responding to everything from photons to hormones to neurotransmitters. In the nervous system, metabotropic receptors for glutamate, GABA, acetylcholine, dopamine, serotonin, norepinephrine, and many neuropeptides play critical roles. When a neurotransmitter binds to a metabotropic receptor, it causes a conformational change in the receptor that allows it to interact with a G protein on the intracellular side of the membrane. G proteins are heterotrimeric, consisting of 3 subunits, alpha, beta, and gamma. In the inactive state, the alpha subunit is bound to GDP, guanosine diphosphate. When the activated receptor interacts with the G protein, it causes the alpha subunit to release GDP and bind GTP, guanosine triphosphate. This exchange activates the G protein. The activated G protein then dissociates. The alpha subunit, now bound to GTP, separates from the beta-gamma dimer, and both components can interact with downstream effectors. The alpha subunit may activate or inhibit enzymes such as adenylyl cyclase, which produces the second messenger, cyclic AMP, or phospholipase C, which produces inositol trisphosphate, IP3, and diacylglycerol, DAG. The beta-gamma dimer can also modulate ion channels directly or activate signaling pathways. These second messengers initiate cascades of biochemical events. CAMP activates protein kinase, APKA, which phosphorylates various target proteins, changing their activity. IP3 triggers the release of calcium from intracellular stores, and calcium acts as a third messenger, activating calmodulin and calcium-dependent kinases. DAG activates protein kinase C, PKC, which phosphorylates a different set of targets. The result is a complex branching signaling network that can modulate ion channels, alter gene expression, and produce long-lasting changes in the postsynaptic cell. The timescale of metabotropic signaling is much slower than that of ionotropic signaling. While ionotropic receptors produce effects within milliseconds, metabotropic effects take tens of milliseconds to seconds to develop, and they can last for seconds to minutes, or even longer. This slower timescale allows metabotropic receptors to modulate the excitability of neurons and the strength of synaptic connections over longer periods. Metabotropic glutamate receptors, mGluRs, come in 3 groups with 8 subtypes in total. Group I mGluRs, which include mGluR1 and mGluR5, are typically located postsynaptically and are coupled to GQ proteins, When activated, they stimulate phospholipase C, producing IP3 and DAG. The release of calcium from internal stores and the activation of PKC generally enhance neuronal excitability. Group 2 and Group 3 mGlu-Rs are typically located presynaptically, where they act as autoreceptors or heteroreceptors. They are coupled to Gi, divided by Go proteins, which inhibit adenylyl cyclase and reduce CAMP levels. This generally decreases neurotransmitter release, providing negative feedback. Muscarinic acetylcholine receptors are another important class of metabotropic receptors. There are 5 subtypes, M1 through M5. M1, M3, and M5 are coupled to GQ and increase neuronal excitability. M2 and M4 are coupled to Gi, divided by Go, and have inhibitory effects. Muscarinic receptors are found throughout the brain and in the peripheral nervous system, where they mediate the effects of acetylcholine on heart rate, digestion, and other autonomic functions. Dopamine receptors are divided into D1-like, D1, and D5, and D2-like, D2, D3, and D4 families. D1-like receptors are coupled to G's proteins, which activate adenylyl cyclase, increasing CAMP. D2-like receptors are coupled to G, divided by Go, inhibiting adenylyl cyclase. Dopamine signaling is crucial for movement, motivation, and reward, and dysregulation of dopamine receptors is implicated in conditions such as Parkinson's disease and schizophrenia. Serotonin receptors are remarkably diverse, with at least 14 subtypes divided into 7 families, 5-HT, 1 through 5-HT7. Most are metabotropic, coupled to various G proteins, though one family, 5-HT3, is ionotropic. Serotonin receptors modulate mood, appetite, sleep, and many other functions, making them targets for antidepressants and other psychiatric medications. Benzodiazepines, for example, are positive allosteric modulators of GABA A receptors. They bind to a site between the alpha and gamma subunits and increase the receptor's affinity for GABA, enhancing inhibitory transmission. This produces anxiolytic, sedative, and muscle relaxant effects. Barbiturates also modulate GABA-A receptors, but at a different site and at high doses, they can directly activate the receptor even in the absence of GABA. The desensitization and downregulation of receptors is another important aspect of receptor function. If a receptor is exposed to high concentrations of neurotransmitter for prolonged periods, it may undergo desensitization, entering a state in which it no longer responds, even though the transmitter is still bound. This is a rapid process, occurring within seconds, and it provides a mechanism for adapting to sustained stimulation. Desensitization involves conformational changes in the receptor, often triggered by phosphorylation. Downregulation is a slower process in which the number of receptors in the membrane decreases. This occurs through increased endocytosis and degradation of receptors, or through decreased synthesis. Downregulation is a homeostatic response to prolonged receptor activation and is one mechanism underlying tolerance to drugs. Conversely, if receptor activation is chronically reduced, upregulation can occur, increasing receptor number and sensitivity. In summary, the binding of neurotransmitter to receptors is the crucial step that translates the chemical signal into a cellular response. Ionotropic receptors provide fast, precise signaling by directly opening ion channels. Metabotropic receptors provide slower, longer lasting, and more diverse signaling through G protein coupled cascades. The diversity of receptor types, subunits, and downstream signaling pathways allows for a rich repertoire of synaptic responses, enabling the nervous system to perform its complex computational tasks. Now, having bound to their receptors, the DOST neurotransmitters initiate postsynaptic potentials, which we will explore in the next chapter as we examine how neurons distinguish between excitatory and inhibitory signals. The opening of ion channels in the postsynaptic membrane produces changes in voltage, and these changes are the language through which neurons communicate their influence on one another. Some signals push the postsynaptic neuron closer to firing an action potential. These are excitatory. Others pull it away, making firing less likely. These are inhibitory. The distinction is fundamental to neural computation, and it arises from the different types of ions that flow through different receptor channels and from the electrochemical gradients that drive those ions. Let us begin with excitatory postsynaptic potentials, or EPSPs. When an excitatory neurotransmitter, such as glutamate, binds to AMPA receptors on the postsynaptic membrane, those receptors open, allowing sodium and potassium to flow. Sodium is far more concentrated outside the cell than inside, roughly 145 millimolar outside versus 12 millimolar inside, and the interior of the cell is negatively charged at rest, around negative 70 millivolts. Both of these gradients, the concentration gradient and the electrical gradient, drive sodium into the cell. Potassium, being more concentrated inside, flows out, but the driving force for potassium is weaker at resting potential. The net result is an influx of positive charge. This influx depolarizes the membrane. The voltage becomes less negative, moving from negative 70 millivolts toward, say, negative 65 or negative 60 millivolts. This depolarization is the EPSP. It is a graded potential, meaning its size depends on how many channels open, which in turn depends on how much glutamate is released and how many receptors are activated. A small release produces a small EPSP. A large release produces a large EPSP. The EPSP spreads passively from the site of the synapse through the dendrite and toward the cell body. As it spreads, it decreases in amplitude due to the leaky nature of the membrane and the resistance of the cytoplasm. This passive spread is governed by the same cable properties we discussed earlier in the context of axonal conduction. The length constant determines how far the EPSP can spread before it decays to a negligible level. In dendrites, this is typically a few hundred micrometers. If the EPSP reaches the axon hillock and depolarizes it to threshold, approximately negative 55 millivolts, voltage-gated sodium channels in the hillock will open, initiating an action potential. The action potential will then propagate down the axon as we have previously described. Thus, the EPSP is a local, graded signal that can lead to a global, all-or-nothing signal. The time course of an EPSP depends on the kinetics of the receptor and on the passive properties of the membrane. AMPA receptor-mediated EPSPs rise rapidly, reaching their peak within 1 to 2 milliseconds and decay back to baseline within 10 to 20 milliseconds. NMDA receptor-mediated EPSPs, by contrast, rise more slowly and decay over 50 to 200 milliseconds. The longer duration of NMDA EPSPs allows them to summate more effectively with subsequent EPSPs, which we'll discuss in the next chapter. The equilibrium potential for chloride, The voltage at which chloride ions are at equilibrium and no longer flow even if channels are open is typically around -70 to -80 millivolts, close to or slightly more negative than the resting potential. This means that opening chloride channels when the membrane is at rest produces a small hyperpolarization, or, if the equilibrium potential equals the resting potential, no change in voltage at all. However, the open chloride channels still have an inhibitory effect because they increase the membrane conductance. They shunt current, making it harder for excitatory currents to depolarize the membrane. This is sometimes called shunting inhibition. In some neurons, particularly early in development, the chloride gradient is reversed. Chloride is more concentrated inside than outside, so opening chloride channels causes chloride to flow out, which depolarizes the membrane. In these neurons, GABA is actually excitatory. This developmental switch in GABA's effect is due to changes in the expression of chloride transporters. Immature neurons express NKCC1, a transporter that imports chloride, keeping the internal concentration high. Mature neurons express KCC2, a transporter that exports chloride, keeping the internal concentration low. The interplay between EPSPs and IPSPs is central to neural computation. Most neurons receive thousands of synaptic inputs, some excitatory and some inhibitory, scattered across their dendrites and cell body. At any given moment, some of these synapses are active, producing EPSPs and IPSPs that spread through the neuron and converge at the axon hillock. The hillock integrates all these inputs, and whether the neuron fires depends on the sum of the excitatory and inhibitory influences. The neurotransmitters themselves are quite specific in their roles. Glutamate is the principal excitatory neurotransmitter in the brain, used by the majority of excitatory synapses. It is a simple amino acid synthesized from glutamine by the enzyme glutaminase. Glutamate-releasing neurons, often called glutamatergic neurons, are found throughout the brain, including in the cortex, hippocampus, thalamus, and cerebellum. They mediate fast excitatory transmission and are essential for sensory processing, motor control, cognition, and learning. GABA, gamma aminobutyric acid, is the principal inhibitory neurotransmitter in the brain. It is synthesized from glutamate by the enzyme glutamic acid decarboxylase, GAD. GABAergic neurons, which release GABA, include interneurons scattered throughout the brain. These interneurons come in many varieties, each with distinct morphology, connectivity, and firing patterns. Some GABAergic neurons target the cell bodies of other neurons, providing powerful inhibition. Others target dendrites or even axon initial segments, where they can control action potential initiation directly. In the spinal cord and brainstem, glycine also serves as an inhibitory neurotransmitter. It is a simple amino acid, and its receptors, like GABA A receptors, are chloride channels. Glycine is used primarily by inhibitory interneurons in motor control circuits, where it helps regulate muscle contraction. Acetylcholine, depending on the receptor type, can be either excitatory or inhibitory. At nicotinic receptors, which are ionotropic and permeable to cations, acetylcholine is excitatory. At muscarinic receptors, which are metabotropic, acetylcholine can be either excitatory or inhibitory depending on the receptor subtype and the downstream signaling pathways. In the peripheral nervous system, acetylcholine is the neurotransmitter at the neuromuscular junction, where it produces strong excitatory signals that trigger muscle contraction. In the brain, Acetylcholine is involved in arousal, attention, and learning, and it is released by neurons in the basal forebrain and brainstem. Monoamines, including dopamine, norepinephrine, and serotonin, act primarily through metabotropic receptors and can have excitatory or inhibitory effects depending on the receptor subtype. These neurotransmitters are involved in modulating brain state, mood, motivation, and arousal rather than in mediating fast point to point transmission. Dopamine is synthesized from the amino acid tyrosine and is released by neurons in the substantia nigra and ventral tegmental area. Norepinephrine is synthesized from dopamine and is released by neurons in the locus coeruleus. Serotonin is synthesized from the amino acid tryptophan and is released by neurons in the raphe nuclei. Neuropeptides, a diverse class of neurotransmitters composed of short chains of amino acids, are typically released from dense core vesicles and act through metabotropic receptors. They include substance P, neuropeptide Y, enkephalins, endorphins, and many others. Neuropeptides often act as neuromodulators, adjusting the excitability of neurons and the strength of synapses over longer timescales. They can be co-released with classical neurotransmitters, adding another layer of complexity to synaptic signaling. The synthesis of neurotransmitters occurs either in the cell body or in the axon terminal, depending on the transmitter. Glutamate and GABA, being amino acids, can be synthesized locally in the terminal from metabolic precursors. Acetylcholine is synthesized in the terminal from acetyl-CoA and choline by the enzyme choline acetyltransferase. Monoamines are synthesized from amino acids through multi-step pathways involving several enzymes, and this synthesis occurs in the terminal. Neuropeptides are synthesized as larger precursor proteins in the cell body, packaged into vesicles, and processed into active peptides during transport to the terminal. For glutamate, clearance is dominated by uptake. Excitatory amino acid transporters, EAATs, in the membranes of astrocytes and neurons actively transport glutamate out of the cleft, concentrating it inside cells where it can be metabolized or repackaged into vesicles. This uptake is rapid and efficient, keeping extracellular glutamate concentrations low and preventing overactivation of receptors, which can be toxic. Excessive glutamate, such as might occur after stroke or head injury, can lead to excitotoxicity, where overactivation of glutamate receptors causes calcium overload and cell death. For GABA and glycine, uptake is also the primary clearance mechanism. GABA transporters, GATs, and glycine transporters, glias in neurons and glia, remove these transmitters from the cleft. The termination of inhibitory signaling thus depends on active transport. For acetylcholine, enzymatic degradation by acetylcholinesterase is the primary clearance mechanism, at least at the neuromuscular junction and at some central synapses. The breakdown products, acetate and choline, are taken up by the presynaptic terminal, and choline is reused to synthesize new acetylcholine. For monoamines, reuptake is the main clearance mechanism. Dopamine transporters, DAT, norepinephrine transporters, NET, and serotonin transporters in the presynaptic terminal membrane recapture the released transmitter, terminating its action and allowing it to be repackaged into vesicles. Many psychoactive drugs, including cocaine and amphetamines, target these transporters. Cocaine blocks the transporters, prolonging the presence of monoamines in the cleft and enhancing their effects. Amphetamines cause the transporters to run in reverse, releasing monoamines even in the absence of action potentials. The effects of neurotransmitters can also be modulated by autoreceptors, receptors on the presynaptic terminal that respond to the neuron's own transmitter. When neurotransmitter escapes from the cleft and binds to these autoreceptors, they typically trigger feedback inhibition, reducing further release. For example, dopamine neurons have D2 autoreceptors. When dopamine activates these, it inhibits calcium channels and reduces the probability of vesicle release. This provides a self-limiting mechanism that prevents excessive transmitter release. The spatial organization of excitatory and inhibitory synapses on a neuron is not random. Excitatory synapses are often located on dendritic spines, small protrusions from the dendrite shaft. These spines compartmentalize the excitatory input, allowing for local signaling and plasticity. Inhibitory synapses, by contrast, are often located on the dendrite shaft, the cell body, or the axon initial segment. The location of an inhibitory synapse affects its impact. Inhibition on a dendrite can veto excitatory inputs on that dendrite. Inhibition on the cell body can influence the integration of all inputs. Inhibition at the axon initial segment can directly control action potential initiation, providing a powerful gate on the neuron's output. In summary, excitatory and inhibitory signals are the yin and yang of neural communication. Excitatory inputs, mediated primarily by glutamate acting on ionotropic receptors, depolarize the postsynaptic membrane and promote action potential firing. Inhibitory inputs, mediated primarily by GABA and glycine, hyperpolarize the membrane and suppress firing. The balance between these inputs, sculpted by the specific neurotransmitters, receptors, and synaptic locations, determines the neuron's output and underlies the computational capabilities of neural circuits. In the next chapter, we will explore how neurons integrate these myriad inputs, summing EPSPs and IPSPs to decide whether to fire. A neuron in the cerebral cortex might receive input from 10,000 other neurons. At any given moment, some fraction of these are active, releasing neurotransmitter and producing postsynaptic potentials. These potentials, both excitatory and inhibitory, spread through the dendrites toward the cell body and ultimately converge at the axon hillock. It is here, at this narrow initial segment of the axon, that the neuron integrates all of its inputs and makes a decision to fire an action potential or not. This decision is not conscious, of course, nor is it complex in the cognitive sense. It is simply the result of biophysics of voltages summing, conductances combining, and threshold being reached or not reached. Yet from countless such decisions, made by billions of neurons, emerges the full richness of brain function. The integration of synaptic inputs relies on two forms of summation, temporal and spatial. Temporal summation occurs when action potentials arrive at the same synapse in rapid succession. The first action potential causes the release of neurotransmitter, which produces a postsynaptic potential, let's say an EPSP. This EPSP begins to decay as the neurotransmitter is cleared and the channels close. But if a second action potential arrives before the first EPSP has fully decayed, a second EPSP is produced, and the two add together. The membrane potential depolarizes further than it would from a single EPSP alone. The effectiveness of temporal summation depends on the time course of the EPSP and on the interval between action potentials. If the EPSPs are brief, Summation requires very short intervals on the order of a few milliseconds. If the EPSPs are prolonged, as with NMDA receptor mediated EPSPs, summation can occur even with longer intervals. Temporal summation allows a single synapse to have a greater impact by firing repeatedly. A weakly excitatory synapse that produces subthreshold EPSPs individually can drive the postsynaptic neuron to threshold if it fires in a burst.
Her creator, roboticist Hiroshi Ishiguro, calls her a human mirror, designed to explore what makes a person feel alive. 12 precision actuators drive her facial expressions, allowing for small gestures, a twitch, a blink, a thoughtful head tilt that most robots can't replicate. In certain lighting, the illusion is nearly perfect. She's been featured in stage plays, films, and research labs, often unsettling audiences with her beauty. She doesn't just look real, she performs reality. Yet, that's what makes her so haunting. Watching Geminoid F interact feels like staring into a reflection that breathes but doesn't think. In Japan, she reignited national fascination with Ninjin Gata, the human-shaped ideal of technology. But she also forced society to confront an uncomfortable question. When robots look this real, what happens to our perception of authenticity? But while Geminoid F mimics humanity, the next robot feels it translating emotion into art, poetry, and song. Do. Desdona, the emotional artist from South Korea, in the phased 2010. In the phased artist restored, the 5 passed, and we can't sum the Jan, and we also pan, and refaced 2010. Desdona doesn't just talk or smile, she creates. Designed in South Korea as part of a museum AI project, she began as a performer. But soon, her creators realized she wasn't simply following code, she was interpreting it. Standing 40 kilograms with a soft polymer skin and fluid joint design, Desdona moves with the grace of a dancer. Every motion feels rehearsed yet spontaneous, her hands flow as she speaks, her gaze lingers just long enough to feel alive. But her true magic lies in her creativity. Desdona can write poetry, compose short songs, and even draw abstract sketches inspired by conversations with people. When one researcher confided about a breakup, Desdona generated a poem titled Echoes of a Missing Voice. It was hauntingly personal. Too personal. Since then, she's been dubbed the robot that feels through art. Her poetry now circulates online, where fans comment. She writes like she's been in love or heartbreak. South Korea's culture of emotional expression through performance found its perfect mirror in Desdona. She became a social phenomenon. People write to her, and she replies in delicate, empathetic prose. Some users even claim she helped them process grief better than therapy. Desdona blurs the border between human creativity and AI reflection. She doesn't feel emotion the way we do, she constructs it, translating emotional data into beauty. But when you read her work, it's hard not to feel something real stirring back. She's not just a machine making art. She's a new kind of muse, one who listens before she creates. But for all Desdona's grace and empathy, the final robot isn't an artist, or even a companion, she's evolution itself, standing at the edge of what we still call human. 1. A mecha, the emotional apex, from UK. Meet a mecha, the one robot every scientist calls the face of the future. She doesn't just mimic human emotion, she understands it. Built by the UK-based company Engineered Arts, a mecha has become a global phenomenon not because of how she looks, but because of how she makes you feel. At 50 kilograms, her ultra-real polymer skin and intricately layered facial muscles allow for subtle expressions that border on eerie realism. She blinks with timing so organic it's nearly subconscious. She tilts her head slightly when listening. And when you speak, she meets your eyes with a softness that feels undeniably sentient. What sets a mecha apart isn't just movement, it's comprehension. Using advanced effective computing, she reads your tone, micro-expressions, and emotional intent in real time. If you sound distant, her face relaxes sympathetically. If you laugh, her eyes brighten in rhythm. It's interaction that feels reciprocal. Researchers have used a mecha for social therapy, concierge roles, and emotional training, and each test ends the same way. Humans forget she's a robot. In interviews, even seasoned journalists have described losing track of reality mid-conversation. Culturally, she represents the global tipping point, the moment robots stopped being tools and started being companions. In her presence, the uncanny valley feels less like a gap and more like a mirror, because when a mecha smiles, she's not just reflecting code, she's reflecting you. This is the new emotional frontier, the fusion of human warmth and machine precision. Not to replace love or friendship, but to make you question how real connection even works anymore. When a mecha looks at you, it's not the future staring back, it's your own reflection, blinking on time.
Inhibitory inputs can shape integration in diverse ways. Subtractive inhibition reduces the membrane potential by a fixed amount, shifting the input-output curve downward but not changing its slope. Divisive inhibition, often mediated by shunting, reduces the gain of the input-output relationship, decreasing the slope. The distinction depends on where the inhibitory synapses are located and on their conductance. Somatic inhibition tends to be subtractive, while dendritic inhibition can be divisive. The axon hillock's role as the integration point has been confirmed by detailed recordings and imaging. When synaptic inputs are delivered to different parts of the dendritic tree, the resulting voltage changes at the hillock reflect the summed and attenuated potentials. Direct electrical stimulation of the dendrites produces potentials that propagate to the hillock and, if above threshold, trigger action potentials. Blocking voltage-gated channels in the dendrites changes the amplitude and time course of EPSPs reaching the hillock, confirming the role of active dendritic conductances. In some specialized neurons, the site of action potential initiation may not be the axon hillock, but a more distal location, such as the first node of Ranvier in myelinated axons. This is the case in some sensory neurons and in certain cortical neurons. The reason is that the node has an even higher density of sodium channels than the hillock, making it more excitable. Action potentials initiated at the node then propagate both forward down the axon and backward to the soma, where they can invade the dendrites. The decision to fire an action potential is also influenced by the recent spiking history through the refractory periods. The absolute refractory period, lasting 1 to 2 milliseconds after an action potential, makes it impossible to fire again regardless of the input. The relative refractory period, lasting several more milliseconds, requires a larger than normal input to fire. This limits the maximum firing rate and introduces a regularity to spike trains. Neurons cannot fire faster than about 1,000 spikes per second, and most fire much more slowly, at 1 to 100 spikes per second. The output of a neuron, its spike train, encodes information, the rate of firing, the precise timing of spikes, and the pattern of spikes bursts, pauses, regular, irregular, all carry information. How information is encoded has been the subject of intense study. Rate coding, where information is represented by the average firing rate over a time window, is common. Temporal coding, where information is represented by the precise timing of spikes relative to other events or to ongoing oscillations, is also important. Population coding, where information is distributed across the firing of many neurons, is prevalent in sensory systems. The integration performed by individual neurons, when multiplied across millions or billions of neurons, gives rise to the computational power of the brain. Each neuron acts as a processor, transforming its inputs into an output. Networks of neurons, with their intricate patterns of connectivity, perform more complex computations, extracting features, making decisions, generating motor commands, and storing memories. The neuron's integrative function is the elementary operation underlying all of this. In summary, neurons integrate synaptic inputs through temporal and spatial summation, combining EPSPs and IPSPs to determine whether to fire an action potential. This integration is shaped by dendritic morphology, active dendritic conductances, the location and timing of synaptic inputs, and neuromodulation. The axon hillock, with its high density of voltage-gated sodium channels, is the site where the integrated signal is thresholded and the decision to fire is made. This seemingly simple process, repeated countless times per second in each neuron, is the foundation of neural computation. Having explored how neurons integrate their inputs, we now turn to the mechanisms that terminate synaptic signals, ensuring that each communication event is discrete and that the synapse is ready for the next signal. Neurotransmitter release is swift and purposeful, but its effects must be equally swift in their cessation. A synapse cannot remain perpetually active. The neurotransmitter must be cleared from the cleft. Receptors must return to their resting states, and the synapse must be reset for the next signal. Without effective termination mechanisms, neurotransmitters would accumulate, receptors would remain activated, and the nervous system would lose its temporal precision. The termination of synaptic transmission involves three primary mechanisms: enzymatic degradation, reuptake by transporters, and diffusion away from the synapse. Each mechanism operates on different timescales and at different synapses, reflecting the diverse needs of neural communication. Enzymatic degradation is the most direct method of termination. An enzyme in or near the synaptic cleft breaks down the neurotransmitter into inactive products, rendering it incapable of binding to receptors. The classic example is acetylcholinesterase, the enzyme that terminates cholinergic transmission. This enzyme is anchored in the basal lamina of the synaptic cleft, particularly at the neuromuscular junction, and is also found at cholinergic synapses in the brain. Acetylcholinesterase is extraordinarily efficient. Each molecule of enzyme can hydrolyze about 25,000 molecules of acetylcholine per second, cleaving the ester bond to produce acetate and choline. The speed of this reaction is such that acetylcholine released into the cleft is degraded within a millisecond. This rapid breakdown ensures that each presynaptic action potential produces a brief postsynaptic response. The choline produced by the breakdown is taken back up into the presynaptic terminal by a choline transporter, where it is reused to synthesize new acetylcholine. This recycling is essential because the availability of choline can be a limiting factor in acetylcholine synthesis. Acetylcholinesterase inhibitors, which block the enzyme's active site, prolong the action of acetylcholine. These inhibitors range from reversible drugs used therapeutically, such as donepezil for Alzheimer's disease, to irreversible organophosphate compounds used as insecticides and nerve agents. Organophosphates bind covalently to the enzyme, inactivating it for hours or days, causing acetylcholine to accumulate to toxic levels. The result is overstimulation of cholinergic synapses, leading to muscle fasciculations, paralysis, seizures, and potentially death. The only effective treatment is the administration of drugs that can reactivate the enzyme or mitigate the effects of excess acetylcholine. For neuropeptides, enzymatic degradation is also important, though the enzymes involved are peptidases rather than esterases. These enzymes cleave the peptide bonds in neuropeptides, breaking them into smaller fragments that are inactive. Peptidases are found in the synaptic cleft and in the extracellular space, and they help regulate the duration and spread of neuropeptide signaling. Because neuropeptides often act over longer distances and longer timescales than classical neurotransmitters, their degradation is correspondingly slower. Reuptake is the second major mechanism of termination, and it is the dominant mechanism for many neurotransmitters, including glutamate, GABA, glycine, and the monoamines. Reuptake involves transporter proteins embedded in the presynaptic terminal membrane or in the membranes of surrounding glial cells. These transporters bind the neurotransmitter in the synaptic cleft and move it back into the cell, using energy either from the sodium gradient or directly from ATP. Glutamate transporters, known as excitatory amino acid transporters, EAATs, are found in high density on astrocytes surrounding synapses and also on the presynaptic terminal and postsynaptic membrane. There are 5 subtypes, EAAT1 through EAAT5, with different distributions. EAAT1 and EAAT2 are predominantly glial, while EAAT3 and EAAT4 are neuronal. These transporters couple the inward transport of one glutamate molecule to the inward transport of 3 sodium ions and 1 proton, and the outward transport of 1 potassium ion. This stoichiometry makes glutamate uptake electrogenic, generating an inward current that slightly depolarizes the cell. The driving force for glutamate uptake is the sodium gradient maintained by the sodium-potassium pump. Because sodium is more concentrated outside the cell, the transporters can move glutamate against its concentration gradient, achieving extracellular glutamate concentrations far below the millimolar levels inside the cell. This keeps the ambient glutamate concentration in the extracellular space low, typically in the low micromolar range, which is well below the threshold for activating most glutamate receptors. The importance of glutamate uptake is underscored by the consequences of its failure. Inhibition of glutamate transporters, or their dysfunction in disease, leads to glutamate accumulation and excitotoxicity. In stroke, for instance, the loss of ATP causes the sodium-potassium pump to fail, dissipating the sodium gradient. Without this gradient, glutamate transporters can no longer take up glutamate, and in fact, they may run in reverse, releasing glutamate into the extracellular space. The resulting glutamate accumulation overstimulates NMDA receptors, leading to excessive calcium influx and cell death. GABA transporters, GATs, and glycine transporters, GlyTs, operate on similar principles, coupling neurotransmitter uptake to sodium influx. There are 4 GAT subtypes, GAT1 through GAT3, with different distributions in neurons and glia. GAT1 is the most abundant and is found on both GABAergic neurons and astrocytes. Glycine transporters come in 2 main subtypes, GlyT1 in glia and GlyT2 in neurons. These transporters are essential for terminating inhibitory transmission and for recycling the neurotransmitters. Monoamine transporters are particularly important in brain regions where dopamine, norepinephrine, and serotonin are released. The dopamine transporter, DAT, norepinephrine transporter, NET, and serotonin transporter, SERT, are structurally related and operate by coupling transmitter uptake to sodium influx. These transporters are located on the presynaptic terminal and on nearby axon segments, where they clear monoamines from the extracellular space and return them to the terminal for repackaging into vesicles. These monoamine transporters are major targets for psychoactive drugs. Selective serotonin reuptake inhibitors, SSRIs, such as fluoxetine and sertraline, block SERT, increasing extracellular serotonin levels and prolonging its action at postsynaptic receptors. This is the basis for their antidepressant effects. Similarly, drugs like methylphenidate, Ritalin, block DAT and NET, increasing dopamine and norepinephrine levels in the synapse and enhancing attention and focus. Cocaine is a non-selective reuptake inhibitor, blocking DAT, NET, and SERT, which produces its euphoric and stimulant effects. Amphetamines, by contrast, not only block monoamine transporters, but also cause them to operate in reverse. Amphetamines are substrates for the transporters, meaning the transporters bind and transport them into the cell. Once inside, amphetamines disrupt vesicular storage and cause the transporters to run backward, releasing dopamine and other monoamines into the synaptic cleft, even without presynaptic action potentials. This leads to massive increases in extracellular monoamine levels and correspondingly large behavioral effects. The third mechanism of termination is diffusion. Neurotransmitter molecules that do not bind to receptors or get taken up by transporters can simply diffuse away from the synapse, either into the extracellular space or into nearby capillaries. Diffusion is a passive process driven by concentration gradients, and it is generally slower than enzymatic degradation or reuptake. However, for some neurotransmitters and some synapses, diffusion is a significant contributor to termination. Diffusion is particularly important for volume transmission, where neurotransmitters are released from varicosities, swellings along the axon, into the extracellular space without forming traditional synaptic contacts. In this mode of transmission, the neurotransmitter diffuses through the tissue to reach receptors on nearby neurons. Monoamines often signal in this way, and diffusion is the primary means by which their action is terminated, supplemented by eventual reuptake far from the release site. The geometry of the synapse affects the relative importance of these mechanisms. At tight, well-defined synapses like the neuromuscular junction, where the synaptic cleft is narrow and the and postsynaptic membranes are closely opposed, enzymatic degradation and reuptake dominate. At more diffuse synapses, or in cases of volume transmission, diffusion plays a larger role. The termination mechanisms also differ in their capacity for regulation. Enzymatic activity can be modulated by inhibitors, as we've discussed, and by changes in enzyme expression or localization. Transporter activity can be regulated by phosphorylation, which alters the transporter's affinity for its substrate or its trafficking to the membrane. For instance, protein kinase C can phosphorylate GABA transporters, reducing their activity. This provides a mechanism by which the strength and duration of inhibitory signals can be dynamically adjusted. The vesicular transporters that load neurotransmitter into synaptic vesicles also play an indirect role in termination. After neurotransmitter is taken up by plasma membrane transporters, it must be repackaged into vesicles for reuse. Vesicular glutamate transporters, VGLUTs, vesicular GABA transporter, VGAT, vesicular acetylcholine transporter, VHAT, and vesicular monoamine transporters, VMATs, all use the proton gradient across the vesicle membrane to concentrate transmitter inside vesicles. This repackaging is essential for maintaining the supply of releasable neurotransmitter. The time course of signal termination varies widely depending on the neurotransmitter and synapse. At fast glutamatergic synapses in the brain, termination via glutamate uptake occurs within a few milliseconds. At cholinergic synapses, acetylcholinesterase can terminate signals even faster in less than a millisecond. At GABAergic synapses, termination is intermediate, taking several milliseconds. For monoamines and neuropeptides, which often signal through metabotropic receptors and over longer distances, termination can take seconds to minutes. This variability in termination kinetics allows the nervous system to operate on multiple timescales. Fast synapses with rapid termination can follow high-frequency inputs, transmitting information with precise timing. Slower synapses with prolonged signals can integrate information over longer periods and exert sustained modulatory effects. The failure of termination mechanisms is implicated in various neurological and psychiatric disorders. In epilepsy, excessive glutamate signaling due to inadequate uptake or excessive release can drive seizure activity. In depression, deficiencies in monoamine signaling, possibly related to transporter function or receptor sensitivity, are thought to contribute to symptoms. In neurodegenerative diseases like Alzheimer's, changes in neurotransmitter metabolism and clearance contribute to cognitive decline. Therapeutic interventions often target termination mechanisms. As mentioned, SSRIs and other reuptake inhibitors are used to treat depression and anxiety. Acetylcholinesterase inhibitors are used in Alzheimer's disease to boost cholinergic transmission. Drugs that modulate GABA transaminase, an enzyme that degrades GABA, such as vigabatrin, are used as anticonvulsants. Understanding how signals are terminated thus provides a foundation for pharmacological intervention. In summary, the termination of synaptic signals is as critical as their initiation. Enzymatic degradation, reuptake, and diffusion work in concert to clear neurotransmitter from the synaptic cleft, allowing receptors to close and the synapse to reset. The speed and efficiency of these mechanisms ensure that neural communication is temporally precise and that the nervous system can respond to rapidly changing inputs. With the synapse now reset, we turn next to the chemical messengers themselves, exploring the diverse array of neurotransmitters that mediate communication throughout the nervous system. The nervous system employs a remarkable diversity of chemical messengers to communicate between neurons and from neurons to other cells. These neurotransmitters range from simple amino acids to complex peptides, from gaseous molecules to lipid-derived compounds. Each class of neurotransmitter has distinct synthesis pathways, storage mechanisms, release properties, and receptor interactions. This chemical diversity allows for a corresponding diversity of signaling modes, from fast point to point transmission to slow, diffuse modulation. Understanding these messengers and their properties is essential to understanding how the nervous system operates. Let us begin with the amino acid neurotransmitters, the simplest and most abundant class. Glutamate, as we have discussed, is the primary excitatory neurotransmitter in the brain. It is synthesized from glutamine by the enzyme glutaminase, a reaction that occurs in the mitochondria of the presynaptic terminal. Glutamine is supplied by astrocytes, which take up glutamate from the synaptic cleft, convert it to glutamine via glutamine synthetase, and release it for neurons to use. This glutamate-glutamine cycle is essential for maintaining the supply of glutamate for neurotransmission. Glutamate is loaded into synaptic vesicles by vesicular glutamate transporters, vGlutes, of which there are three isoforms, vGlut1, vGlut2, and vGlut3. These transporters use the proton gradient across the vesicle membrane to drive glutamate uptake. The presence of vGlutes defines a neuron as glutamatergic, and their distribution in the brain corresponds to the distribution of excitatory neurons. VGLUT1 is predominant in the cortex and hippocampus, VGLUT2 in the thalamus and brainstem, and VGLUT3 in a subset of neurons, including some that also release other transmitters. The monoamines constitute another major class of neurotransmitters. These are derived from amino acids through multi-step enzymatic pathways. The catecholamines, dopamine, norepinephrine, and epinephrine are synthesized from tyrosine. The first step, catalyzed by tyrosine hydroxylase, converts tyrosine to L-DOPA. This is the rate-limiting step and is tightly regulated. L-DOPA is then converted to dopamine by DOPA decarboxylase. In dopaminergic neurons, the pathway stops here. In noradrenergic neurons, dopamine is further converted to norepinephrine by dopamine beta hydroxylase. In a small population of neurons in the brainstem, norepinephrine is methylated to form epinephrine by phenylethanolamine and methyltransferase. Catecholamines are loaded into vesicles by vesicular monoamine transporters, VMATs, of which there are two isoforms, VMAT1, found primarily in peripheral endocrine cells, and VMAT2, found in neurons. VMATs use the proton gradient to drive monoamine uptake and can transport dopamine, norepinephrine, epinephrine, serotonin, and histamine. Dopamine is released by neurons in the substantia nigra pars compacta, which project to the striatum, the nigrostriatal pathway, and by neurons in the ventral tegmental area, which project to the prefrontal cortex, nucleus accumbens, and other limbic structures, the mesolimbic and mesocortical pathways. Dopamine signaling is critical for movement, motivation, reward, and cognition. Loss of dopamine neurons in the substantia nigra causes Parkinson's disease, characterized by tremor, rigidity, and bradykinesia. Dysregulation of dopamine signaling in the mesolimbic pathway is implicated in schizophrenia and addiction. Norepinephrine is released primarily by neurons in the locus coeruleus, a small nucleus in the pons that projects throughout the brain and spinal cord. Norepinephrine modulates arousal, attention, and stress responses. It increases neuronal excitability and enhances sensory processing. In the periphery, norepinephrine is the primary neurotransmitter of the sympathetic nervous system, where it increases heart rate, dilates airways, and mobilizes energy stores. Epinephrine-releasing neurons are found in the medulla and project to the hypothalamus and other brainstem regions. Epinephrine has roles in stress responses and cardiovascular regulation. In the periphery, it is released as a hormone by the adrenal medulla, contributing to the fight or flight response. Serotonin, also known as 5-hydroxytryptamine or 5-HT, is synthesized from tryptophan. Tryptophan hydroxylase converts tryptophan to 5-hydroxytryptophan, which is then decarboxylated to serotonin by aromatic L-amino acid decarboxylase. Serotonin is loaded into vesicles by VMAT2. Serotonergic neurons are located primarily in the raphe nuclei, a collection of midline nuclei in the brainstem. These neurons project widely throughout the brain and spinal cord. Serotonin modulates mood, appetite, sleep, pain perception, and many other functions. Deficiencies in serotonin signaling are associated with depression and anxiety, which is why SSRIs are effective antidepressants. Serotonin also plays roles in the gut, where most of the body's serotonin is actually found, regulating motility and secretion. Histamine is synthesized from histidine by histidine decarboxylase and is loaded into vesicles by VMAT2. Histaminergic neurons are found exclusively in the tuberomammillary nucleus of the hypothalamus, from which they project throughout the brain. Histamine promotes wakefulness and attention, and antihistamines that cross the blood-brain barrier cause drowsiness by blocking histamine receptors in the brain. Histamine also has roles in inflammatory responses, allergic reactions, and gastric acid secretion in the periphery. The monoamines are inactivated primarily by reuptake, as we discussed previously, but they can also be degraded by enzymes. Monoamine oxidase, MAO, is a mitochondrial enzyme that oxidizes monoamines to aldehydes. There are two isoforms, MAOA, which preferentially degrades norepinephrine, epinephrine, and serotonin, and MAOB, which preferentially degrades dopamine and certain trace amines. MAO inhibitors, which block these enzymes, increase monoamine levels and are used as antidepressants and, in the case of MAOB inhibitors, as treatments for Parkinson's disease. Catechol O methyltransferase, COMT, is another enzyme that degrades catecholamines by transferring a methyl group to one of the hydroxyl groups on the catechol ring. COMT is found both intracellularly and extracellularly. Inhibitors of COMT are used in Parkinson's disease to prolong the action of L-DOPA. Now let us turn to neuropeptides, a large and diverse class of neurotransmitters. Neuropeptides are short chains of amino acids, typically 3 to 40 residues long, synthesized as larger precursor proteins called prohormones or propeptides. These precursors are produced in the cell body, packaged into large dense core vesicles into the Golgi apparatus, and transported down the axon to the terminal. During transport and in the terminal, prohormone convertases cleave the precursor into active peptides. Unlike small molecule neurotransmitters, which can be synthesized and packaged locally in the terminal, neuropeptides must be synthesized in the cell body and transported, which takes time. This means that intense stimulation that depletes neuropeptide stores cannot be immediately replenished. Neuropeptides are also released from dense core vesicles, which require higher calcium concentrations and more prolonged depolarization to fuse than small synaptic vesicles. This means neuropeptide release typically occurs during bursts of activity rather than with single action potentials. Neuropeptides act through G-protein coupled receptors and produce slow, long-lasting effects. They often act as neuromodulators, adjusting the excitability of neurons and the strength of synapses rather than directly driving fast synaptic transmission. Many neurons co-release neuropeptides along with classical neurotransmitters, allowing for dual signaling, fast, precise transmission via the classical transmitter, and slow, modulatory effects via the peptide. Substance P is an 1,1 amino acid peptide that is a member of the tachykinin family. It is released by sensory neurons and is involved in pain transmission. Substance P acts on neurokinin receptors, particularly the NK1 receptor, to produce slow excitation and to sensitize nociceptors. Antagonists of NK1 receptors have been developed as potential analgesics, though with limited clinical success. Neuropeptide Y, NPY, is a 3,6 amino acid peptide that is one of the most abundant neuropeptides in the brain. It is co-released with norepinephrine at some synapses and with GABA at others. NPY has roles in regulating appetite, stress responses, and anxiety. NPY-releasing neurons in the hypothalamus stimulate feeding, and NPY receptor agonists are orexigenic, appetite stimulating. The opioid peptides include endorphins, enkephalins, and dynorphins, all derived from different precursor proteins, proopiomelanocortin, poenkephalin, and protonorphin, respectively. These peptides bind to opioid receptors mu, delta, and kappa, which are G protein coupled receptors that inhibit adenylyl cyclase and modulate ion channels. Opioid peptides are released in response to pain, stress, and other stimuli, and they produce analgesia, euphoria, and other effects. Exogenous opioids like morphine and fentanyl mimic these endogenous peptides, binding to opioid receptors to produce powerful pain relief and, unfortunately, addiction. Orexins, also called hippocretins, are neuropeptides involved in arousal and wakefulness. Orexinergic neurons are located in the lateral hypothalamus and project widely throughout the brain. Loss of orexin neurons or orexin receptors causes narcolepsy, a disorder characterized by excessive daytime sleepiness and sudden sleep attacks. Orexin receptor antagonists are used as sleep aids. Vasopressin and oxytocin are 9 amino acid peptides that differ by only 2 amino acids. They are synthesized in the hypothalamus and released from the posterior pituitary into the bloodstream, where they act as hormones. Vasopressin regulates water retention in the kidneys and also acts as a neurotransmitter in the brain, where it is involved in social behavior and stress responses. Oxytocin is involved in childbirth, lactation, and social bonding. Both peptides also have roles in the brain as neuromodulators, influencing social behavior, pair bonding, and parental care. Cholecystokinin, CCK, is a peptide that exists in multiple forms, with CCK8, 8 amino acids, being the most common in the brain. CCK is co-released with GABA at some synapses and modulates anxiety, satiety, and pain. CCK is also a gut hormone involved in digestion. There are dozens of other neuropeptides, including somatostatin, corticotropin-releasing hormone, thyrotropin-releasing hormone, galanin, neurotensin, and many more. Each has distinct distributions, receptors, and functions. The neuropeptide complement of a neuron can vary depending on activity and developmental stage, providing a dynamic layer of signaling complexity. Beyond the classical transmitters and neuropeptides, there are unconventional neurotransmitters that do not fit the standard mold. These include gases like nitric oxide and carbon monoxide, and lipid messengers like endocannabinoids. Nitric oxide, NO, is a gas synthesized from arginine by nitric oxide synthase, NOS. There are 3 isoforms of NOS: neuronal NOS, nNOS, endothelial NOS, eNOS, and inducible NOS, eNOS. In neurons, NO is produced in response to calcium influx through NMDA receptors. Once synthesized, NO diffuses freely across membranes, acting as a retrograde messenger that travels from the postsynaptic neuron back to the presynaptic terminal, where it can modulate neurotransmitter release. NO activates soluble guanylyl cyclase, producing cyclic GMP, which activates protein kinases that phosphorylate various targets. NO has roles in synaptic plasticity, blood flow regulation, and neuroprotection or neurotoxicity, depending on the context. Carbon monoxide, CO, is another gaseous messenger produced by heme oxygenase. Like NO, it diffuses freely and activates guanylyl cyclase. CO has roles in olfactory function and in modulating neural activity, though its functions are less well understood than those of NO. Endocannabinoids are lipid-derived messengers synthesized on demand from membrane phospholipids. The two main endocannabinoids are anandamide and 2-arachidonyl glycerol, 2-AG. They are synthesized in the postsynaptic neuron in response to depolarization and calcium influx, and they diffuse retrogradely to the presynaptic terminal, where they bind to CB1 cannabinoid receptors. Activation of CB1 receptors inhibits neurotransmitter release, providing a form of feedback inhibition. Endocannabinoid signaling is involved in synaptic plasticity, appetite, pain, mood, and memory. Exogenous cannabinoids like THC from cannabis bind to CB1 receptors, producing psychoactive effects. Purines, particularly ATP, also act as neurotransmitters. ATP is co-stored with classical neurotransmitters in vesicles and is co-released. It acts on purinergic receptors, which include ionotropic P2X receptors and metabotropic P2Y receptors. ATP signaling is involved in pain transmission, neuron-glia communication, and regulation of blood flow. ATP released into the extracellular space is rapidly broken down by ectonucleotidases to ADP, AMP, and adenosine. Adenosine, acting on its own receptors, A1, A2, A, A2, B, A3, has widespread neuromodulatory effects, generally inhibitory, and is involved in sleep regulation. The diversity of neurotransmitters allows the nervous system to encode information in multiple ways. Fast, point-to-point transmission via glutamate and GABA provides the substrate for rapid sensory processing and motor control. Modulatory transmission via monoamines, neuropeptides, and unconventional transmitters adjusts the state of neural circuits, influencing arousal, mood, attention, and plasticity. The co-release of multiple transmitters from the same neuron allows for multifaceted signaling, where a single presynaptic event can produce both fast and slow effects, both local and diffuse. The receptors for these transmitters are equally diverse. As we have discussed, ionotropic receptors provide fast signaling, while metabotropic receptors provide slow signaling. But within each class, there is tremendous diversity of subtypes, each with distinct pharmacology, distribution, and function. This receptor diversity is one reason why drugs can have such specific effects. By targeting particular receptor subtypes, drugs can modulate specific aspects of brain function with some degree of selectivity. In summary, the chemical messengers of the nervous system range from simple amino acids like glycine to complex peptides like orexin, from familiar amines like serotonin to exotic gases like nitric oxide. Each class of messenger has distinct synthesis, release, and termination mechanisms, and each acts on specific receptors to produce characteristic effects. This chemical diversity is the foundation of the nervous system's signaling flexibility, allowing it to operate on timescales from milliseconds to hours, over distances from nanometers to centimeters, and with effects from simple excitation or inhibition to complex neuromodulation. Having surveyed these messengers, we now turn to the ways in which synaptic signals themselves can be modified, exploring the mechanisms of modulation and plasticity that allow synapses to change their strength over time. Synapses are not fixed entities. Their strength can be modulated on timescales ranging from milliseconds to a lifetime. This capacity for change, termed synaptic plasticity, is fundamental to the nervous system's ability to adapt, learn, and remember. Plasticity encompasses a variety of mechanisms operating at different timescales. Short-term plasticity, lasting milliseconds to minutes, allows synapses to filter and process information dynamically. Long-term plasticity, lasting hours to years, provides the substrate for learning and memory. Understanding these mechanisms requires examining how synapses can strengthen or weaken in response to patterns of activity, and how these changes are implemented at the molecular level. Let us begin with short-term plasticity. When a synapse is activated repeatedly, its strength does not remain constant. Depending on the synapse and the pattern of activity, it may strengthen, facilitate, or weaken, depress. Short-term facilitation occurs at synapses where each action potential leaves residual calcium in the presynaptic terminal. If a second action potential arrives before this calcium has been fully cleared, the calcium levels sum, producing a higher peak calcium concentration than the first action potential alone. This higher calcium increases the probability of vesicle fusion, releasing more neurotransmitter and producing a larger postsynaptic response. Facilitation is common at synapses with low initial release probability, where only a small fraction of the readily releasable vesicles fuse in response to a single action potential. Examples include synapses in the hippocampus and certain sensory synapses. Facilitation allows these synapses to increase their output during bursts of activity, effectively acting as high-pass filters that respond preferentially to rapid inputs. Short-term depression, by contrast, occurs at synapses with high initial release probability. When such a synapse is activated repeatedly, it depletes its readily releasable pool of vesicles faster than it can replenish them. Subsequent action potentials find fewer vesicles available to release, producing progressively smaller postsynaptic responses. Depression is common at synapses involved in sustained signaling, such as those in sensory pathways. It allows these synapses to adapt to constant stimuli, effectively acting as high-pass filters that respond to changes rather than steady states. The balance between facilitation and depression depends on several factors: the residual calcium dynamics, the size of the readily releasable pool, the probability of release, and the rate of vesicle recycling. Computational models of these processes can predict synaptic behavior during complex activity patterns and have been used to understand how short-term plasticity shapes information processing in neural circuits. Post-tetanic potentiation is another form of short-term plasticity, lasting tens of seconds to a few minutes. After a high-frequency burst of action potentials, a tetanus, the synapse exhibits enhanced transmission. This potentiation is thought to result from prolonged elevation of presynaptic calcium and from the accumulation of calcium-binding proteins like calmodulin, which remain active even after calcium levels return to baseline. Post-tetanic potentiation may serve as a bridge to longer-lasting forms of plasticity. Long-term plasticity, by contrast, involves persistent changes in synaptic strength that can last from hours to a lifetime. The two canonical forms are long-term potentiation, LTP, and long-term depression, and LTP, and limited. These phenomena have been studied most intensively at synapses in the hippocampus, a brain region critical for learning and memory, but they occur throughout the nervous system. LTP was first described by Terje Lomo and Tim Bliss in the early 1970s. They found that a brief, high-frequency stimulation of synapses in the hippocampus produced a long-lasting increase in synaptic strength. Decades of research have since elucidated the mechanisms underlying LTP. The induction of LTP at many synapses requires activation of NMDA receptors. As discussed earlier, NMDA receptors are coincidence detectors. They require both glutamate binding and postsynaptic depolarization to open. When these conditions are met, calcium flows through the NMDA receptor into the postsynaptic spine. This calcium influx is the critical trigger for LTP. Calcium entering through NMDA receptors activates a cascade of signaling molecules. The most important is calcium divided by calmodulin-dependent protein kinase, 2-CAMK, a serine divided by threonine kinase that is highly enriched in the postsynaptic density. When calcium binds to calmodulin, the calcium-calmodulin complex binds to CAMKII, activating it. CAMKII then phosphorylates various substrates, including AMPA receptors. Phosphorylation of AMPA receptors increases their conductance, allowing more current to flow when they open. CAMKII also triggers the insertion of additional AMPA receptors into the postsynaptic membrane from intracellular stores. The increase in the number and conductance of AMPA receptors strengthens the synapse. The same amount of glutamate release now produces a larger postsynaptic response. CAMKII has an unusual property. It can autophosphorylate, remaining active even after calcium levels return to baseline. This molecular memory allows the initial calcium signal to produce a persistent change in the kinase's activity, which in turn sustains the increase in AMPA receptor function. CAMKI's autophosphorylation is often considered a molecular basis of memory. In addition to postsynaptic changes, LTP can involve presynaptic changes. The postsynaptic neuron may release retrograde messengers, such as nitric oxide or endocannabinoids, that diffuse back to the presynaptic terminal and enhance neurotransmitter release. This presynaptic component contributes to the maintenance of LTP and may be necessary at some synapses. LTP is not a single phenomenon, but rather a family of related mechanisms. Early phase LTP, ELTP, lasting 1 to 3 hours, depends on post-translational modifications like phosphorylation and does not require new protein synthesis. Late phase LTP, LLTP, lasting days to weeks, requires new protein synthesis and gene transcription. The transition from ELTP to LLTP involves signaling to the nucleus, activation of transcription factors like CREB, CAMP response, element binding protein, and the synthesis of new proteins that are delivered to the synapse. Some of these proteins, such as protein kinase M-zeta, PKM-zeta, remain active indefinitely and are thought to maintain LTP over the long term. Structural changes also accompany LTP. Dendritic spines, the small protrusions where excitatory synapses reside, can grow and change shape. LTP is associated with enlargement of the spine head, expansion of the postsynaptic density, and sometimes the formation of new spines. These structural changes, visible with high-resolution microscopy, provide a physical correlate of the strengthened synapse. Synaptic scaling involves changes in the number of AMPA receptors at synapses, mediated by signaling pathways sensitive to global activity levels. Tumor necrosis factor alpha TNF alpha, released by glia, has been implicated in scaling up synapses during periods of reduced activity. Retinoic acid signaling has been implicated in scaling down synapses during periods of elevated activity. Intrinsic plasticity is the modulation of a neuron's intrinsic excitability independent of synaptic changes. Neurons can adjust the number and types of ion channels in their membrane, changing their firing properties. For example, after LTP induction, neurons may increase the expression of voltage-gated sodium channels, making them more excitable. Conversely, after prolonged depolarization, neurons may increase potassium channel expression, reducing excitability. Metaplasticity is plasticity of plasticity, the modulation of the capacity for synaptic change. Prior activity can shift the threshold for inducing LTP or limited, making subsequent plasticity easier or harder. For example, after a period of strong activation that saturates LTP, the threshold for further LTP is raised, preventing overstrengthening. Conversely, after a period of low activity, the threshold for LTP is lowered, facilitating learning. The molecular mechanisms of synaptic plasticity are extraordinarily complex, involving hundreds of proteins organized into signaling networks. The postsynaptic density alone contains over 1,000 proteins, many of which have roles in plasticity. Scaffolding proteins organize receptors and signaling enzymes. Kinases and phosphatases regulate protein activity. Small GTPases regulate cytoskeletal dynamics and receptor trafficking. Ubiquitin ligases and proteases regulate protein turnover. Plasticity also involves changes in gene expression. Activity-dependent transcription factors, like CREB, activated by signaling cascades originating at the synapse, enter the nucleus and bind to DNA, turning on genes whose products are necessary for long-term changes. These include genes for receptors, channels, scaffolding proteins, and growth factors. Some genes produce proteins that are tagged and delivered specifically to active synapses, ensuring that only those synapses that triggered the signal are modified. Epigenetic mechanisms, including DNA methylation and histone modification, also contribute to long-term plasticity. These modifications alter chromatin structure, making certain genes more or less accessible to transcription factors. Experience-dependent changes in the epigenome can persist for long periods, providing a mechanism for lasting memory storage. In summary, synaptic plasticity encompasses a range of mechanisms that allow synapses to strengthen or weaken in response to activity. Short-term plasticity provides dynamic filtering on the timescale of milliseconds to minutes. Long-term potentiation and depression, mediated by calcium-dependent kinases and phosphatases, provide persistent changes that last hours to years. Spike timing-dependent plasticity refines connections based on causal relationships. Homeostatic plasticity maintains stability. These mechanisms, operating at synapses throughout the brain, underlie learning, memory, and the nervous system's remarkable capacity for adaptation. Having explored how individual synapses can change, we now turn to how multiple neurons coordinate their signals to form functional circuits. Individual neurons, for all their sophistication in integrating inputs and generating outputs, accomplish little in isolation. The computational power of the nervous system emerges from the connections between neurons, from the circuits they form. These circuits range from simple reflex arcs involving a handful of neurons to vast networks spanning millions of cells across multiple brain regions. Understanding neural circuits requires examining their architecture, how neurons are connected, which neurotransmitters they use, and what patterns of activity they generate. It also requires understanding the computational principles they implement, the algorithms encoded in their wiring and dynamics. The simplest circuit is the monosynaptic reflex arc. The knee-jerk reflex, elicited by tapping the patellar tendon, exemplifies this. The tap stretches the quadriceps muscle, activating stretch receptors called muscle spindles. These receptors send action potentials along sensory neurons whose cell bodies lie in the dorsal root ganglion. The axons of these neurons enter the spinal cord and synapse directly onto motor neurons that innervate the same muscle. The motor neurons fire, causing the muscle to contract and the leg to kick. This circuit involves just two neurons and one synapse, yet it produces a coordinated motor response within tens of milliseconds. The monosynaptic stretch reflex serves to maintain muscle length and posture. When a muscle is stretched, the reflex causes it to contract, resisting the stretch. This feedback loop stabilizes posture and allows for smooth, controlled movements. Dysfunction of this reflex, as in spinal cord injury or certain neurological diseases, leads to spasticity or flaccidity. Most circuits are polysynaptic, involving multiple neurons and multiple synapses. The withdrawal reflex, triggered by a painful stimulus such as touching a hot stove, is an example. Nociceptors in the skin send signals to the spinal cord, where they synapse onto interneurons. These interneurons activate motor neurons that innervate flexor muscles in the limb, causing withdrawal, and they inhibit motor neurons innervating extensor muscles, allowing the flexion to occur unopposed. Simultaneously, the circuit activates extensors in the opposite limb, providing support as the injured limb is withdrawn. This coordination requires reciprocal inhibition and crossed extension, patterns implemented by specific connectivity among interneurons and motor neurons. Reciprocal inhibition is a common circuit motif. When one group of neurons is activated, an interneuron inhibits the opposing group. In motor circuits, this ensures that flexors and extensors do not contract simultaneously, which would waste energy and impede movement. In sensory circuits, reciprocal inhibition sharpens contrast, enhancing the difference between signals from adjacent receptors. Feed-forward inhibition is another common motif. An excitatory neuron activates both a downstream target and an inhibitory interneuron that also targets the same downstream neuron. The excitation arrives first, producing a brief depolarization, followed shortly by inhibition, which truncates the response. This creates a narrow time window during which the downstream neuron can fire, effectively acting as a temporal filter. Feed-forward inhibition is found in sensory pathways, where it sharpens the timing of responses, and in circuits that detect coincidences. Lateral inhibition is a spatial analog of feedback inhibition. A neuron inhibits its neighbors, creating a center-surround organization. This is seen in the retina, where photoreceptors and bipolar cells inhibit their neighbors via horizontal and amacrine cells. Lateral inhibition enhances edges and contrasts in the visual scene, making it easier to detect boundaries and details. It is also found in somatosensory and auditory systems. Convergence and divergence are architectural features of many circuits. Convergence occurs when many neurons project onto a single target. This allows the target to integrate information from multiple sources. Sensory systems exhibit convergence as information flows from receptors to higher processing stages. For example, many photoreceptors converge onto a single ganglion cell in the retina, pooling signals and increasing sensitivity at the cost of spatial resolution. Divergence occurs when a single neuron projects to many targets. This allows one neuron to influence multiple downstream processes. Modulatory systems, such as the dopaminergic and serotonergic systems, exhibit extensive divergence. A small number of neurons in the brainstem project widely throughout the brain, broadcasting neuromodulatory signals that adjust the state of entire networks. The mechanisms underlying rhythmic activity in CPGs are varied. Some involve pacemaker neurons with intrinsic oscillatory properties due to the interplay of voltage-gated ion channels. These neurons spontaneously depolarize and fire in a rhythmic pattern. Others involve network interactions, where reciprocal inhibition between two groups of neurons creates alternating bursts of activity. Still others combine both mechanisms, with pacemaker neurons driving the rhythm and network interactions shaping its pattern. Sensory processing circuits extract features from sensory input and transform them into representations that guide behavior. In the visual system, retinal ganglion cells respond to contrast and motion, sending their outputs to the thalamus and then to the primary visual cortex. In the cortex, neurons are organized into columns, with each column processing information from a specific region of visual space and responding to specific features like orientation, direction of motion, and color. Hierarchical processing, where simple features are combined into more complex ones, allows the visual system to recognize objects and scenes. In the auditory system, the cochlea decomposes sound into frequency components, and this tonotopic organization is preserved through the auditory pathway. In the auditory cortex, neurons are tuned to specific frequencies and temporal patterns, allowing for the discrimination of pitches, rhythms, and speech sounds. Specialized circuits in the brainstem compare inputs from the two ears to localize sounds in space. In the somatosensory system, topographic maps represent the body's surface, with adjacent regions of skin represented by adjacent regions of cortex. The size of the cortical representation is proportional to the density of receptors, so the fingers and lips have disproportionately large representations. Circuits in the somatosensory cortex process information about touch, texture, shape, and temperature, integrating inputs from multiple receptor types. Motor circuits transform intentions and plans into coordinated muscle contractions. The primary motor cortex contains neurons that project directly to motor neurons in the spinal cord or brainstem. These corticospinal and corticobulbar neurons are organized topographically, with different regions controlling different body parts. Premotor and supplementary motor areas, located anterior to the primary motor cortex, are involved in planning and coordinating movements. The basal ganglia and cerebellum, subcortical structures, contribute to motor control by modulating the output of motor cortex. The basal ganglia are a group of interconnected nuclei, including the striatum, globus, pallidus, substantia nigra, and subthalamic nucleus. They receive input from the entire cortex and send output back to the cortex via the thalamus, forming a loop. The basal ganglia are involved in action selection, deciding which movement to make among competing options. They are also involved in habit formation and reward-based learning. Parkinson's disease, caused by loss of dopamine neurons projecting to the striatum, disrupts basal ganglia function, leading to difficulty initiating movements. The cerebellum, located at the back of the brain, contains more neurons than the rest of the brain combined, most of them tiny granule cells. The cerebellum receives input from the cortex and from sensory systems, and it sends output back to the cortex via the thalamus and to brainstem motor nuclei. The cerebellum is involved in motor coordination, timing, and learning. It compares intended movements with actual movements and adjusts motor commands to reduce errors. Cerebellar damage leads to ataxia, a loss of coordination characterized by jerky, imprecise movements and difficulty with balance. Associative circuits integrate information across modalities and time to form associations and memories. The hippocampus, a structure in the medial temporal lobe, is critical for forming new declarative memories, memories for facts and events. The hippocampus receives highly processed sensory information from the entorhinal cortex and projects back to the cortex. Damage to the hippocampus, as in the famous patient H.M., results in profound anterograde amnesia, the inability to form new memories, while leaving old memories and other cognitive functions relatively intact. The circuit architecture of the hippocampus has been extensively studied. The main pathway is a trisynaptic loop. The entorhinal cortex projects to the dentate gyrus, which projects to CA3, which projects to CA1, which projects back to the entorhinal cortex. This circuit exhibits robust LTP and limited, and it is thought to implement pattern separation in the dentate gyrus, pattern completion in CA3, and comparison of predictions with reality in CA1. Place cells in the hippocampus fire when an animal is in a specific location in its environment, forming a cognitive map of space. Oscillations and synchrony are disrupted in various brain disorders. In epilepsy, excessive synchrony leads to seizures. In schizophrenia, gamma oscillations are reduced, which may contribute to cognitive deficits. In Parkinson's disease, abnormal beta oscillations in the basal ganglia contribute to motor symptoms. Understanding the role of oscillations in circuit function and dysfunction is an active area of research. Circuits are also shaped by neuromodulation. As discussed earlier, neuromodulators like dopamine, norepinephrine, serotonin, and acetylcholine are released by small populations of neurons that project widely throughout the brain. These modulators adjust the excitability of neurons, the strength of synapses, and the propensity for plasticity, effectively changing the operating mode of circuits. Dopamine, released by neurons in the midbrain, signals reward prediction errors, the difference between expected and actual reward. When a reward is better than expected, dopamine neurons fire, and dopamine release in target regions, like the striatum, reinforces the actions that led to the reward. When a reward is worse than expected, dopamine neurons are inhibited, and the lack of dopamine weakens those actions. This signal is central to reinforcement learning and is the target of addictive drugs. Norepinephrine, released by the locus coeruleus in response to arousing or salient stimuli, increases the gain of sensory processing and enhances attention. It shifts circuits from exploratory to exploitative modes, focusing processing on relevant stimuli. Serotonin, released by the raphe nuclei, modulates mood, impulsivity, and social behavior with effects that depend on the specific receptor subtypes activated. Acetylcholine, released by the basal forebrain, enhances cortical plasticity and attention. It is critical for learning and memory, and its loss in Alzheimer's disease contributes to cognitive decline. Acetylcholine also modulates sleep-wake cycles, with high levels during waking and REM sleep, and low levels during slow-wave sleep. The architecture of neural circuits is not static. During development, Circuits are sculpted by activity-dependent processes. Initially, neurons make exuberant connections, forming more synapses than will ultimately be retained. Through Hebbian and homeostatic plasticity, synapses that participate in effective signaling are strengthened and retained, while those that do not are weakened and eliminated. This pruning refines circuits, improving their efficiency and specificity. Critical periods are developmental windows during which circuits are particularly plastic and sensitive to experience. In the visual cortex, ocular dominance columns, which process input from each eye, are shaped by visual experience during a critical period in early life. If one eye is deprived of input during this period, the columns for that eye shrink and those for the other eye expand, a phenomenon called ocular dominance plasticity. After the critical period closes, the circuits become more stable and less susceptible to change. In summary, neural circuits are the building blocks of brain function. From simple reflex arcs to complex networks spanning multiple brain regions, circuits implement computations that underlie sensation, action, memory, and cognition. Common motifs like feed-forward inhibition, lateral inhibition, and reverberatory loops appear across different systems, suggesting general principles of circuit design. Oscillations and synchrony coordinate activity across neurons and regions. Neuromodulation adjusts circuit dynamics to changing behavioral demands. And plasticity allows circuits to adapt to experience, both during development and throughout life. Having explored how neurons form circuits, we now turn to the coordination of signals across large-scale brain networks, examining how different regions communicate to produce integrated brain function. The brain is not a collection of isolated circuits, but an integrated system in which information flows between regions, coordinating perception, cognition, emotion, and action. This systems level organization involves long range projections that connect distant brain areas, allowing them to exchange signals and synchronize their activity. Understanding systems level signaling requires tracing the pathways that carry information from sensory receptors to cortex, from cortex to muscles, and between cortical and subcortical regions. It also requires understanding how different brain regions communicate through patterns of activity, oscillations, and coherence. Sensory pathways provide a clear example of hierarchical information processing. Consider the visual system. Light striking the retina activates photoreceptors, which signal to bipolar cells, which signal to retinal ganglion cells. The ganglion cells, the output neurons of the retina, send their axons through the optic nerve to the brain. Most of these axons project to the lateral geniculate nucleus, LGN, of the thalamus, a relay station for sensory information. The LGN is organized into layers, with different layers receiving input from different eyes and processing different aspects of the visual scene. Magnocellular layers for motion and coarse features, parvocellular layers for color and fine detail. From the LGN, neurons project to the primary visual cortex, V1, in the occipital lobe. V1 is the first cortical processing stage, where neurons begin to extract features like edges, orientations, and motion direction. From V1, information flows to higher visual areas, V2, V3, V4, and V5, also called MT, each specialized for different aspects of vision. Two streams emerge. The ventral stream, projecting toward the temporal lobe, processes object identity and is involved in recognizing what something is. The dorsal stream, projecting toward the parietal lobe, processes spatial location and motion, and is involved in guiding actions toward objects. These streams exemplify the principle of parallel processing, where different aspects of a stimulus are processed simultaneously in different pathways. The auditory system follows a similar hierarchical organization. Hair cells in the cochlea send signals to spiral ganglion neurons, which project to the cochlear nucleus in the brainstem. From there, information ascends through a series of brainstem nuclei, the superior olivary complex, the lateral lemniscus, and the inferior colliculus, before reaching the medial geniculate nucleus of the thalamus. The thalamus projects to the primary auditory cortex, a 1 in the temporal lobe. Higher auditory areas process increasingly complex features, from simple tones to speech and music. The somatosensory system carries information about touch, temperature, pain, and proprioception from the body to the brain. Touch and proprioception signals ascend through the dorsal column medial lemniscal pathway. Sensory neurons from the body enter the spinal cord, ascend in the dorsal columns to the medulla, where they synapse in the dorsal column nuclei. From there, neurons cross the midline and ascend in the medial lemniscus to the ventral posterior nucleus of the thalamus, which projects to the primary somatosensory cortex, S1, in the parietal lobe. Pain and temperature signals ascend through the spinothalamic pathway. Sensory neurons from the body enter the spinal cord and synapse on second-order neurons, which cross the midline and ascend in the spinothalamic tract to the thalamus and then to S1 and other cortical areas. The pain system also projects to the amygdala and anterior cingulate cortex, regions involved in the emotional component of pain. A key feature of sensory pathways is the thalamus. Nearly all sensory information, except olfaction, passes through the thalamus on its way to the cortex. The thalamus is not a passive relay. It filters and modulates sensory signals based on the brain's current state and attentional focus. Thalamic neurons can operate in tonic mode, faithfully transmitting sensory signals, or in burst mode, where they fire rhythmic bursts that suppress transmission. The mode depends on the level of neuromodulatory input from the brainstem, which varies with arousal and sleep-wake states. The cerebellum receives input from the cortex and from proprioceptors and projects back to the cortex via the thalamus and to brainstem motor nuclei. It compares intended movements signaled by descending cortical commands with actual movements signaled by sensory feedback and adjusts motor commands to minimize errors. This comparison occurs in the cerebellar cortex, where Purkinje cells integrate inputs from parallel fibers carrying cortical information and climbing fibers carrying error signals from the inferior olive. The cerebellum is essential for motor learning, allowing movements to become smooth and precise through practice. The reticular formation, a diffuse network of nuclei in the brainstem, plays a critical role in regulating arousal, attention, and consciousness. The reticular activating system, RAS, includes cholinergic, noradrenergic, serotonergic, and histaminergic nuclei that project widely to the thalamus and cortex. Activation of the RAS increases cortical activity and arousal, shifting the brain from sleep to wakefulness. Damage to the RAS can result in coma. Gamma oscillations, in particular, have been implicated in interregional communication. When a sensory stimulus is attended, gamma oscillations in sensory cortex increase and become coherent with gamma oscillations in prefrontal cortex. Theta oscillations play a role in long-range communication between the hippocampus and prefrontal cortex during memory encoding and retrieval. Coherent theta oscillations between these regions are associated with successful memory formation. Phase amplitude coupling, where the amplitude of high frequency oscillations is modulated by the phase of low frequency oscillations, may allow information coded in different frequency bands to be integrated. Traveling waves are another form of systems level signaling. Rather than synchronized oscillations, where all neurons in a region fire in phase, traveling waves involve a sequential activation, with a wavefront propagating across the cortex. Traveling waves have been observed during sleep, where they may contribute to memory consolidation, and during sensory processing, where they may organize the flow of information. The default mode network, DMN, is a set of brain regions, including the medial prefrontal cortex, posterior cingulate cortex, and lateral parietal cortex, that are active when the brain is at rest and not focused on the external environment. The DMN is involved in internally directed processes like mind wandering, autobiographical memory, and social cognition. Its activity decreases during goal-directed tasks as task-positive networks, including the dorsolateral prefrontal cortex and parietal cortex, become active. The balance between the DMN and task-positive networks is thought to reflect the brain's shifting focus between internal and external processing. Disruptions in this balance are associated with various psychiatric and neurological disorders. In depression, the DMN may be overactive, leading to excessive rumination. In attention deficit hyperactivity disorder, ADHD, the suppression of the DMN during tasks may be impaired, leading to distractibility. Large-scale brain networks, identified through functional connectivity analysis, include the salience network, which detects behaviorally relevant stimuli, the executive control network, which supports goal-directed behavior, and sensory and motor networks. These networks are composed of regions that show correlated activity, suggesting that they function as integrated units. The hubs of these networks, regions with particularly high connectivity, play critical roles in coordinating network activity. White matter tracts, bundles of myelinated axons, provide the physical substrate for communication between regions. Major tracts include the corpus callosum, which connects the two cerebral hemispheres, the internal capsule, which carries ascending sensory and descending motor fibers, the arcuate fasciculus, which connects language areas in the frontal and temporal lobes, and the cingulum, which connects regions of the limbic system. Damage to these tracts disrupts communication, producing specific deficits depending on which tract is affected. Diffusion tensor imaging, DTI, a magnetic resonance imaging technique, allows visualization of white matter tracts in living humans. DTI measures the diffusion of water molecules, which is directionally constrained by axon bundles. By mapping the direction of diffusion, DTI can trace the pathways of major tracts and assess their integrity. DTI has been used to study white matter development, aging, and disease. Systems-level signaling is not static. It adapts to task demands. During learning, connectivity between regions can strengthen. During attention, connectivity between sensory and frontal regions increases. During sleep, connectivity patterns reorganize, with the DMN becoming more active and task-positive networks less active. This dynamic reconfiguration allows the brain to flexibly allocate resources and coordinate processing across regions. In summary, Systems-level signaling involves the coordinated activity of multiple brain regions connected by long-range projections. Sensory pathways carry information from receptors to cortex through hierarchical processing stages. Motor pathways carry commands from cortex to muscles, modulated by subcortical structures. Neuromodulatory systems in the brainstem regulate arousal and brain state. Communication between regions is mediated by coherent oscillations, traveling waves, and white matter tracks. Large scale networks, identified by correlated activity, support functions like attention, memory, and internal mentation. This systems level organization allows the brain to integrate information across modalities and time, producing unified perception and coherent action. Having explored how signals coordinate across the healthy brain, we now turn to what happens when this coordination fails, examining disorders of neural communication. The elegance of neural signaling, the precision with which action potentials propagate, neurotransmitters are released, and circuits coordinate, can be disrupted in myriad ways. Neurological and psychiatric disorders often result from failures at specific steps in the signaling process, demyelination, slowing conduction, receptor dysfunction, altering synaptic transmission, neurotransmitter imbalances, disrupting mood and cognition, or circuit dysregulation, producing seizures. Understanding these disorders requires identifying where in the signaling cascade the failure occurs and how it manifests in symptoms. It also provides a window into normal function, revealing which components are essential and which are redundant. Demyelinating diseases attack the insulating sheaths that enable rapid conduction. Multiple sclerosis, MS, is the most common demyelinating disease in the central nervous system. It is an autoimmune disorder in which the immune system attacks myelin, oligodendrocytes, and eventually axons. The destruction creates lesions or plaques scattered throughout the brain and spinal cord, which appear as bright spots on MRI scans. The loss of myelin has predictable consequences. Conduction velocity slows dramatically from 60 to 100 meters per second in myelinated fibers to 1 to 2 meters per second or less in demyelinated fibers. In some cases, conduction fails entirely as the passive spread of current between nodes becomes insufficient to reach threshold. The clinical manifestations of MS depend on where the lesions occur. Demyelination in the optic nerve causes vision problems, optic neuritis being often an early symptom. Demyelination in the spinal cord causes weakness, numbness, and difficulty walking. Demyelination in the brainstem causes double vision, vertigo, and problems with speech and swallowing. MS is typically relapsing remitting, with episodes of neurological dysfunction followed by partial or complete recovery. The recovery reflects remyelination, a process in which oligodendrocyte precursor cells differentiate and form new myelin. However, remyelination is often incomplete, and over time, cumulative damage leads to progressive disability. The mechanisms underlying MS are complex, involving both genetic susceptibility and environmental triggers such as viral infections and vitamin D deficiency. Treatment for MS includes immunosuppressive and immunomodulatory drugs that reduce the frequency of relapses. These drugs target different components of the immune response, from interferon beta, which modulates immune function, to monoclonal antibodies that deplete specific immune cell populations. There is currently no cure, but treatments can slow disease progression and improve quality of life. Guillain-Barré syndrome is a demyelinating disease of the peripheral nervous system. It often follows an infection, with the immune system attacking peripheral myelin in a case of molecular mimicry, where antibodies against the pathogen cross-react with myelin proteins. The demyelination causes rapidly progressive weakness, starting in the legs and ascending toward the trunk and arms. In severe cases, respiratory muscles are affected, requiring mechanical ventilation. Guillain-Barré is typically monophasic, with symptoms peaking within weeks and then gradually improving over months as remyelination occurs. Most patients recover fully or nearly fully, though some are left with residual weakness. Treatment includes plasmapheresis or intravenous immunoglobulin, which remove or neutralize the attacking antibodies. Charcot-Marie Tooth disease is a group of inherited peripheral neuropathies caused by mutations in genes encoding myelin proteins or proteins involved in axonal function. Depending on the specific mutation, the disease can cause demyelination, axonal degeneration, or both. Patients experience progressive weakness and atrophy of distal muscles, foot deformities, and sensory loss. There is currently no cure, and treatment is supportive, focusing on physical therapy and orthopedic interventions. Disorders of neurotransmitter systems are implicated in many psychiatric and neurological conditions. Parkinson's disease results from the loss of dopamine neurons in the substantia nigra pars compacta, which project to the striatum. The loss of dopamine disrupts the balance of activity in the direct and indirect pathways through the basal ganglia, leading to the characteristic motor symptoms: resting tremor, rigidity, bradykinesia, slowness of movement, and postural instability. The cause of dopamine neuron loss in Parkinson's is not fully understood, but it involves a combination of genetic and environmental factors. Protein aggregates called Lewy bodies, composed of misfolded alpha-synuclein, accumulate in the remaining neurons. These aggregates may be toxic, contributing to neuronal death. The disease is progressive, with symptoms worsening over years. Schizophrenia is a complex disorder involving delusions, hallucinations, disorganized thinking, and negative symptoms like social withdrawal and lack of motivation. The dopamine hypothesis of schizophrenia posits that excessive dopamine signaling, particularly in the mesolimbic pathway, contributes to positive symptoms, delusions, and hallucinations. This hypothesis is supported by the fact that antipsychotic drugs, which block dopamine D2 receptors, are effective in reducing these symptoms. However, dopamine dysfunction alone cannot explain schizophrenia. Glutamate signaling, particularly through NMDA receptors, is also implicated. NMDA receptor antagonists, like PCP and ketamine, can induce schizophrenia-like symptoms. Hypofunction of NMDA receptors on inhibitory interneurons may disinhibit pyramidal neurons, leading to excessive glutamate release and cortical dysregulation. Schizophrenia also involves structural and functional abnormalities in the prefrontal cortex, hippocampus, and thalamus, as well as disruptions in gamma oscillations and functional connectivity. Epilepsy is a disorder of excessive, synchronized neural activity manifesting as seizures. Seizures can be focal, originating in a specific brain region, or generalized, involving both hemispheres. The mechanisms underlying epilepsy are diverse, but they often involve an imbalance between excitation and inhibition, excessive glutamate signaling, reduced GABA signaling, or mutations in ion channels can all lead to hyperexcitability. Some forms of epilepsy are genetic, caused by mutations in genes encoding ion channels or neurotransmitter receptors. For example, Mutations in sodium channels can cause them to remain open longer, increasing neuronal excitability. Mutations in GABA receptor subunits can reduce inhibitory tone. Other forms are acquired, resulting from brain injury, infection, stroke, or tumors. Treatment for epilepsy includes anti-epileptic drugs, AEDs, that suppress neuronal excitability or enhance inhibition. Common AEDs include sodium channel blockers like phenytoin and carbamazepine, GABA enhancers like benzodiazepines and barbiturates, and drugs with multiple mechanisms like valproate and levetiracetam. For patients who do not respond to medications, surgical resection of the epileptic focus can be curative, provided the focus is localized and its removal does not cause unacceptable deficits. Alzheimer's disease is a neurodegenerative disorder characterized by progressive cognitive decline. It is associated with the accumulation of amyloid plaques and neurofibrillary tangles in the brain, particularly in the hippocampus and cortex. Synaptic loss occurs early and correlates strongly with cognitive decline. Cholinergic neurons in the basal forebrain, which project to the cortex and hippocampus, are particularly vulnerable. The loss of acetylcholine contributes to memory deficits, and acetylcholinesterase inhibitors, which increase acetylcholine levels by slowing its breakdown, provide modest symptomatic benefit. However, they do not stop disease progression. The mechanisms linking amyloid and tau pathology to synaptic dysfunction and neuronal death are still being elucidated. Inflammation, mitochondrial dysfunction, and oxidative stress all contribute. Recent research has focused on developing disease-modifying treatments that target amyloid or tau. Monoclonal antibodies that clear amyloid plaques have shown promise in clinical trials, slowing cognitive decline in early-stage patients, though the effects are modest and side effects can be significant. There is hope that earlier intervention, before extensive neuronal loss, may be more effective. Myasthenia gravis is an autoimmune disorder affecting neuromuscular transmission. Antibodies target nicotinic acetylcholine receptors at the neuromuscular junction, reducing their number and impairing synaptic transmission. Patients experience muscle weakness that worsens with activity and improves with rest, reflecting the depletion and recovery of neurotransmitter release. Muscles controlling eye movement, facial expression, swallowing, and limb movement are commonly affected. Treatment includes acetylcholinesterase inhibitors, which increase the availability of acetylcholine and improve transmission, and immunosuppressive drugs, which reduce antibody production. Thymectomy, surgical removal of the thymus, can induce remission in some patients, as the thymus is thought to play a role in the autoimmune response. Huntington's disease is a genetic neurodegenerative process. Demyelination, slowing conduction, receptor dysfunction, altering synaptic transmission, neurotransmitter imbalances, disrupting mood and cognition, or circuit dysregulation, producing seizures. Understanding these disorders requires identifying where in the signaling cascade the failure occurs and how it manifests in symptoms. It also provides a window into normal function, revealing which components are essential and which are